ఇంకొక పేజ్ కూడా ఉంది దీని తర్వాత. ఓ అబ్బా, అట్లనా? అందుకనేసేనా? ఉమ్, ఉమ్. అట్లయితే సింగిల్ పేజే పెట్టు. Hello all students, all the NEET aspirants, the future doctors, surgeons, scientists. So let's welcome to NEET 2026 live test paper discussion from Resonance Hyderabad. All these Resonance colleges prestigiously is taking this task to help the students give the solutions and key made by the expert resonance faculties for the students utilization. So by within a few minutes we will be sending you also the entire test paper along with the key and solution, right? So all the faculties subject wise are working for you to entirely make the solution and to present you in a very beautiful, very useful explanations, right? So we are waiting, just a couple of minutes time is there that we are waiting for it. And what NEET actually is, NEET full form you already know it, right? So just a couple of information I will be discussing here. The NEET full form is a national eligibility cum entrance test, right? So for undergraduate programs we are targeting for. It is a single medical entrance examination in India for MBBS, BDS, Ayush and other paramedical courses, right? And who conducts this exam? NEET exam is conducted by National Testing Agency, right, every year. And it replaced most of the state level medical entrance examinations, and all together they are conducting in the form of NEET exam now. It's not just pass or fail, it's about your rank, cutoff and counseling. And NEET exam is conducted for 180 questions are given, and the entire paper is for 720 marks, right? Physics, chemistry, botany and zoology. Botany and zoology together it is 90 marks. So coming to today's botany exam paper, botany plus zoology together I will talk about. So the entire biology paper was the most loving paper of students, and most scoring paper also, right? Everybody agree, I think, right? So this paper, almost 85% of the questions are direct from the NCERT lines, and few of the questions are there indirectly asked, little twisted, that they have indirectly asked the questions. But the majority of the questions, I spoke to couple of students also who were preparing very well through NCERT, that they have done extremely well with the support of a NCERT textbook, right? So almost 90% questions they were able to do it in biology. So that's a very beautiful success. So hope majority of you also might have done very good in biology part, half of the subject, half of the NEET exam, right? Then coming to physics. Physics is the most challenging and time consuming test paper this year also, same like 2025 paper. So some of the questions were theoretical, concept based questions were there, but majoritarily the questions were from the numerical part. And obviously for the NEET student, for the numerical questions a little more time will be taken. So that is the reason why in a Resonance all the batches like a NEET training batches, time management is taken care of, maximum focus, maximum importance, because it is not only on the concept. In a NEET examination, time management is very, very, very crucial, right? So if the one who is completing the botany plus zoology questions in uh, less than like 45 minutes at around, right, 45 minutes to 40 minutes to 45 minutes, they only can take little time into physics and chemistry, which are more challenging questions, as everybody knows this, right? So physics is little lengthier, time consuming paper. So it is moderate to hard. I can classify that too with respect to students' perspective only. The most easiest is biology, then physics is moderate to difficult, and then if you come to chemistry paper, majority of the questions are also from the NCRT concepts, which can be comfortably done by with the help of NCRT concepts, NCRT foundation, right? And there is a very good balance also. The students have told that there is a very good balance between physical chemistry, organic chemistry, and inorganic chemistry. And little in organic chemistry, application based questions came, but overall inorganic chemistry very comfortably they can do it, right? And coming to physical chemistry, little of numerical based, so more time will be definitely taken in uh, physical chemistry related questions, right? So so in the entire paper, right, if you compare with the 2025, so in this 2026 paper, almost all the pattern, the level, the way the questions are asked, the way the questions are designed, almost all they are all same only. So might be like the cutoffs or even the competition is also to the same extent like as in last year. So only 50,000 difference is there, that so no much difference will be seen. So overall, out of 720 marks, we have to say we are collecting the student feedback, that how the students, how much score they are expecting from the questions they have done. One of the student was just saying that, sir, 7, 8 questions from biology I was not able to do. 7, 8 questions were little more tricky, is what she was telling about. So that was a challenge for her. And in physics and chemistry, she was able to do out of 45 questions, chemistry 40 questions, 35 to 40 questions she was telling, and around 30 to be correct, and physics 25 questions she has attempted. And another student was saying overall score that she is getting it around from the questions we have discussed with her. She is staying at her 570, 580 marks she is telling, right? So two only students I was able to talk to them. So remaining we are collecting the information from other students also. But overall it is like we want to present you the solutions, like how the questions can be solved, how the solution to be taken, how in a systematic manner the question should be solved, so that you will find it helpful while giving the upcoming exams also, right? So in this pattern, right, NCRT with taking the feedback from many of the students from the last 1.5 decade, NCRT is one major source, right, for the NEET exam cracking. So almost around 24 lakh students, or 24, about 24 lakh students are giving the exam. So you can understand how much weightage, how much importance decision making NCRT will do it, right? So in today's paper, if you see, the easiest is a biology, then second is as usual chemistry, and third difficult side is physics. And it is the pattern every year students will be finding it in the NEET examination, right? So some of the questions, if I remember, some students told that the questions came from ecology, then human physiology, then genetics, modern physics, electrodynamics, like a different electrochemistry, coordination compounds, one of the most important chapter, coordination compounds. And one more student has told like the questions in chemistry are not spread across all the chapters. That was a little surprising for me, because generally it is say we are having we expect that whatever questions are there, every chapter to be given equal importance during the competitive exams. There is no important and unimportant questions. But this time, what that student was he was telling is some chapters questions were given, but some chapter questions were not seen. That I have to cross check it also while by seeing the test paper, because if any chapter is there in the syllabus, definitely we feel like in NTA pattern question, like in a JEE main, they will touch each and every chapter if it is there in the syllabus. That is the reason why in a resonance colleges also, all the chapters will be taken equal importance and will be taken care that every student will be completing every chapter, right now, because we do not know which chapter, from which chapter which question will be coming. So we cannot take the risk also, because it is the student's career, right? So entire 2 year dedicatedly they are preparing, and even some students we are having of a repeater program in MR batch, right? So for them we cannot take any risk. That is why we give the equal importance for each and every chapter. We train them with the worksheets and all. And chemical bonding and coordination chemistry is one area where multiple questions came, is what I can feel from the feedback of the children, right? So in this manner, physics was little, not little actually, it was more time consuming set of questions were there. And in chemistry, moderate questions are there, equal distribution was given, better distribution was there than even 2025 also, is what is told. And biology, as usual, you know, you might have done it very well in biology. So see it, if it is possible, any students have given the exam, you can give in the chat box what score you are expecting, how many questions, total number of questions you have solved subject wise, and the total number of questions, and then how much score you are expecting. And one more thing, you have to understand whether you were able to perform to the level of questions, to the level of performance you thought of before attempting for the exam, right? So these are all some of the things. But anyway, now the exam is over, so you need not take the tension now and right, be very nervous. So that time is over. Now you have to cool down, you have to relax, and then take some rest also, so that whatever the feedback is there, either a positive or a negative side, that can be worked out on a little later after taking some break, right? So from tomorrow onwards, we can work out on it. So today, pleasantly from our expert faculties, listen to the test paper solutions and understand if any of the questions went wrong or you did in a different way, like so the faculty will be giving the proper solution, that they will be very helpful for you, right? Even in the upcoming exam preparation also. Now I, as the time has come now, I welcome our Amulya madam to start with a botany test paper question discussion. Please welcome madam.
Thank you, sir. So, very good evening, dear students. So, I hope you wrote your NEET examination very well. And as sir has already told you, you might have got what is the level of the difficulty, right? So, as I'm here to discuss the first 15 questions of the botany, see, as I've just gone through, as we have gone through the paper, we felt it was quite easy, okay? So, and since most of the questions were directly asked, so I believe, we all believe that you have done your job a great job. So let's begin with the first question. Let us discuss what is the key for these questions. So as you can see, already the name of the chapter also been mentioned here for the quick reference. So the first question says, the evil quartet of biodiversity laws includes which of the following, right? So my dear students, as we know, Nana, so that we have four, right? The evil quartet, quartet ka matlab is four. So we have the four things here. What is that? Overexploitation, alien species invasive, air pollution. So air pollution first is not the option, right? First one is not the option. We'll go to the direct option, which is the correct one. Habitat loss, fragmentation, overexploitation, alien invasiveness, and co-extinction, right? So this is the correct one. Let's go to the next one. Which one of the following is the site for active ribosomal synthesis? So here the ribosomal RNA synthesis It is from the chapter Cell, the Unit of Life. As we know, the nucleolus is the site for the formation of the ribosomal RNA synthesis. Let's move on to the third question. Match the following. So this is from the cell cycle and the cell division, right? So let's look at this option, Nana. G1 phase, S phase, G2 phase and M phase. Okay. So G1 phase, right? The G1 phase, cell is metabolically active and continuously grows but does not replicate its DNA. So A is 2. Let's search for the option. A is 2. So option 1 should be the correct one. Right? S phase, synthesis of the DNA. Right? G2 phase, proteins are synthesized while cell growth continuously. M is the actual cell division. So it is 2, 3, 4, 1. Right? So do we have that? Right, first one is the correct one, right? Let's go to the next one. 2, 3, 4 and D is 1. Okay, D is 1. Let's go to the next one. Match the following again. This is from the ecosystem chapter, right? Let's see, look at this, Nana. What is it? Productivity, net primary productivity, gross primary productivity, secondary productivity. So the productivity, okay, right, productivity, rate of biomass production, that is this one. Net primary productivity, we know gross primary productivity minus respiration, that is B is 1, right? B is 1, B is 1, okay, right? And A is 3, right? Gross primary productivity, right? We have rate of production of organic matter during photosynthesis, and secondary primary productivity, rate of formation of new organic matter by consumer. So the second option is correct. Let's move on to the next one. Which of the following statements are correct? So you can see most of the questions are from the biodiversity and its conservation and the ecosystem also, right? So let's see this now. The Amazon rainforest being cut and cleared for cultivation of soya beans is an example of habitat loss. This is correct. Still, a sea cow and passenger pigeon become extinct due to overexploitation by humans. Absolutely correct. We have to choose the correct statement. The Nile perch introduced into Lake Victoria in Africa helped in population growth. It does not help in population growth. Rather, it is the result. What it is one of the cause of extinction, right? So this is incorrect. Then D, the water hyacinth is an invasive species. Yes, this is also correct. When species becomes extinct, the plant and animal species associated with it are not affected. So this is wrong. So we have to choose the which one? We have to choose the correct statement. Yes, so we have 1, 2, and 3. That is A, B, and D. Let's move on to the next one. How many ATP and NADPH molecules are required to make one molecule of glucose through the caloric pathway? So they asked how many ATPs and NADPH from the chapter photosynthesis. And we know that for one, like to form one glucose, isn't it? The cycle runs for 6 times, है ना? Then for each cycle, we know that 3 ATPs are used and 2 NADPH are spent. So therefore for 6th cycle, it's quite easy, right? 6 threes are 18 ATPs and we have got 6 twos are 12 NADPH. So our answer is the first one. Moving on to the next question. Again, match the following. Right here we have this from the ecosystem chapter. Right. So straight away from this ecosystem chapter, what are the steps from, this is from the decomposition process. Right. So let's see decomposition. Right. Decomposition is breakdown of breakdown of complex organic matter into inorganic substance. So A is 3. Done. Okay, next one. Detritus, dead remains of plants and animals including fecal matter. So B is 4. Done. So mineralization is release of inorganic nutrients by the activity of microbes in the soil. Humification, D is 1. Accumulation of dark colored amorphous colloidal substance. So the correct option is A. A is 3. Okay, so we have got not this one. Then we have B is 4. That is B is 4, right? C is 2. Release of inorganic. Humification. D is 1, right? Let us move on to the next question. In which of the following the ovules are not enclosed by an ovary wall and remains exposed. So straight away the question from the plant kingdom chapter, very easy, right? Which of the following ovules are not enclosed by the ovary wall and remain exposed? So they are, right, exposing. So our correct answer is Pinus, right? So we know that they are talking about the gymnosperms, whereas the others, Laginella, Funaria and Wolffia, they are not the correct suitable options for us. We know the gymnosperms are, right, they have, what do you say, right, exposed seeds. The seeds are naked. Moving on to the next question, right, we have this question from morphology of flowering plants chapter, right? So most of the questions are match the following, है ना? Right, let's look at this. A straight away question from the examples of the placentation, right? Let's talk about this. Marginal, right? Marginal is a P, that is A is 2. Let us search for the answer. Oh, we got, there is only one option which I've got, A is 2. Exile, exile is lemon, right? B is 4, done. Parietal, right? Parietal is, we have got mustard, C is 1, correct. And basil, red basil is marigold, that is D is 3. So our option is option 3, done. Next question. In angiosperms, root hairs arise from which one of the following regions of the root? So very easy question from the chapter Anatomy of the Flowering Plants, right? In angiosperm, root hairs arise from which of the following regions of the root? So the root hairs arises from the region of maturation, right? So the root hairs arises from, we know, right, the root hairs arises from the regions of maturation. This is the root cap in a thimble shape. This is zone of, right, we know, meristematic, है ना? This is zone of meristematic region, right? This is zone of elongation, right? And the final one we have, the last one, that is zone of maturation. Zone of maturation. And from the zone of, or the region of maturation, the root hairs develop, right? That is the question. Now in angiosperm, root hairs arise from which of the following region? Let's move on to the next question. Which one of the following is not a characteristic of plant cells in the phase of elongation, right? So they are asking us from the phase of elongation, that is the region or zone of elongation, which are not the characteristic of these plant cells. So let's talk about that, right? Increased vacuolation, yes, this is the characteristic feature of the elongation. The zone of elongation, the cell starts enlarging, the cell starts elongating and vacuolation takes place. So the first option is correct, but we have to choose which is not the correct option. Let's go to the next one. Large conspicuous nuclei. Yes, they have a large conspicuous nuclei. So this option also is a correct option, right? Cell enlargement, obviously they asked about elongation, so the cell is enlarging, right? New cell wall deposition. New cell wall deposition occurs not in the zone of, what is that, meristematic region, not in the phase of the elongation. So which is not a characteristic feature? This is not a characteristic feature. Our option is this, right? This is not a characteristic feature. So option 4, Nana. Let's move on to the next question. Oh, we have got this question from the molecular basis of inheritance. The statement type of question. It says that which of the following statements are correct with the reference to transcription unit. So let's talk about the transcription unit and a molecular basis, transcription, translation. Right, it's talking about transcription unit. Done. So first point, what do you have to choose? We have to choose the correct statement. A transcription unit in DNA is defined primarily by the regions of promoter, structural and terminator. Absolutely correct. Absolutely correct, right? So the transcription unit, it has got, it has got, right, all these three regions, right? What is it? It has got all these three regions, isn't it? It has got promoter regions, it has got terminator region, this is promoter, this is terminator, and then these are, right, what is the structural genes, right? Structural genes. Done. So the statement 1 is correct. Statement 1 is true. Statement 1 is true. Then second option, the promoter is said to be located towards the 5' end of the structural genes. Yes, this also is absolutely correct, right? So this statement also is correct, right? The promoter is a DNA sequence that provides binding site of RNA polymerase. Obviously, the RNA polymerase responsible for the synthesis of RNA, it comes and binds to the promoter region. This is also correct. Statement 3 also is correct. D, right? The promoter defines the template and coding strands. Obviously, the template strand is fixed, okay, based on the orientation, right? Again, the template and coding strand, this is also absolutely true, right? Then next one, the terminator is located towards the 3' end of the coding strand and defines the end of the process of transcription. Obviously, when the rho factor binds to this region, right, transcription process stops, right? So this statement also is correct. So all A, B, C and D statements are correct. Let us move on to the next one. The main function of bulliform cells in grasses is, right? The question is from anatomy of the flowering plants. The main function of the bulliform cells. Bulliform cells are the cells present in the grasses, isn't it? So we see what happens here, right? So to make the leaf impermeable to fungal spores, absolutely wrong. And these cells basically helps, what is that? To prevent the loss of water, to minimize the loss of water. So as per the statements, if you see, to perform photosynthesis is wrong, to minimize water loss during water stress. Whenever there is a scarcity and deficiency of water, these bulliform cells are responsible for the folding of the leaves, isn't it, to prevent the loss of water. So this is correct. Moving on to the next one. Find out the incorrect statement about photosynthesis from the following. So this is from photosynthesis in higher plants wala chapter, right? Find the incorrect statement. Incorrect statement. Okay, let's see the first statement. The water splitting complex is associated with photosystem 1. No, this is associated with the photosystem 2. We know splitting of water is associated with the photosystem 2, right? So first statement itself is correct. So looking at the options, if you look at here, right, A is incorrect, isn't it? So B only is wrong. We have got only A in one option. Kada? A option is located, or you can see A is present in only one option. Anna. So straight away we can go with this, right? Ikkade ledu, ikkade ledu. But remaining options kuda let us see once. C4 plants use the C3 pathway of CO2 fixation as the main biosynthetic pathway. Obviously true, and we have to choose the incorrect one. Already we have gone through the incorrect one. D kuda, let us check it out. C3 plants exhibit Kranz anatomy. This is wrong because this is a special feature of the C4 plant. So our answer is A and D, right? So let's move on to the next question. Match the following again, right? This also is from the chapter Anatomy of the Flowering Plants. Okay, let's talk about this. Conjunctive tissue, Casparian strips, subsidiary cells, starch sheet, right? So let's talk about this conjunctive tissue. Specialized cells in the vicinity of the guard cell. This is the subsidiary cells, that is the C1. Let us check it out, the C1. Okay, we have got A and right 3. Then we have, what is that? Conjunctive tissue, the tissue between the xylem and the phloem, that is A is 3. A is 3. Okay, this should be the correct answer. Then we have Casparian strips, that is endodermal cells with the suberin deposition, right? That is B is 4, correct? And the starch sheath, the endodermal cells rich in a starch. So if we just check out our answer, A, conjunctive tissue, tissue between xylem and phloem. So A is 3, yes. And B, the Casparian strips, endodermal cells with suberin deposition. B is 4, done. The subsidiary cells, that is C is 1, specialized cells in the vicinity of the guard cell, absolutely correct. And the last one, starch sheath, that is the endodermal cells rich in the starch. So the correct option is D is 2, the correct option is 3, right? So as we have discussed these questions, now I would like to call upon Vikas sir for carrying out the next few questions of the botany.
Yes, hello everyone, good evening, and I hope all of you are doing well. So, today was the NEET exam, 3rd May, and this paper in the biology was very simple and completely from the NCRT. Line to line questions have came from NCRT, a very easy group, and I can say this year the cutoff may rise. Be ready for that. Cutoff will rise this year. Okay, so we'll start with the botany part, the next questions here. So, the first one, so heterophytic development in response to environment is an example of which of the following phenomenon? So what is heterophytic development? So it is a development of different shapes of leaves in the plant. Hetero means different, phylus means leaves. So whenever there is a different development of different shapes of leaves of plant, this is called heterophylous development, and this event is representing the phenomenon of plasticity. इसको plasticity कहते हैं. Next question. In racemose inflorescence. So racemose inflorescence, actually first of all we should know what is actually a inflorescence. Inflorescence is the arrangement of flowers in the floral axis. Okay, so as in the NCRT there are two types of inflorescence. One of the inflorescence is the racemose inflorescence. So what happens here? The main axis of the plant keeps on growing and the flowers are born laterally here. The flowers are born laterally. So in this, the younger flowers are present on the top and the older flowers are present at the bottom. So here you can see these are the young flower on the top part, and these are the old flower in the lower part. Such type of arrangement is called acropetal arrangement. We call this acropetal arrangement. So in the racemose inflorescence, first they are saying the main axis terminates in a flower. No. The main axis keeps on growing. Second, the growth is limited. No. The growth is not limited. The plant will grow continuously and the flowers are born in an acropetal succession. So answer should be option number 3. A very easy question. Now, next one, which of the following disorder is caused by the substitution of glutamic acid by valine at the 6th position of beta globin chain of hemoglobin? So this question is from the principles of inheritance and variation. It is a type of genetic disorder in which you will see that the beta globin chain of hemoglobin is undergoing mutation. So these are some amino acids, the chain of amino acid at the 6th position. Okay, first, second, third, fourth, fifth, sixth. At the 6th position, the glutamic acid is replaced by the valine. Glutamic acid is replaced by the valine. This represents the disease known as sickle cell anemia. It is known as sickle cell anemia. Now going to the next question. So we have a match of the following, and same this question is also from the principles of inheritance and variations. Let's see here. So incomplete dominance example. Incomplete dominance is the inheritance of flower color in antirrhinum. So flower color, there are 3 types of flower color in the antirrhinum, which is also called a dog plant. So here one is a red color flower, one is the white color flower, and one is a pink color flower. Okay, so the incomplete dominance is shown by the pink color here. So A will be second. Co-dominance is the ABO blood group. So in the ABO blood group, the AB blood group shows the phenomenon of co-dominance where both A and B allele express themselves. Okay. Then in the next one, that is pleiotropy, the example will be the phenylketonuria, and polygenic inheritance will be human skin color. Now what is pleiotropy here? So when a single gene show more than one phenotypic effect, that is known as pleiotropic inheritance. And what is polygenic inheritance? When more than one genes control the character or single character, that is called the polygenic inheritance. So match is very easy here. That is A will be 2, okay, B will be 4. So A2, B4 and C will be 3 and D will be 1. Answer should be option number 1 here. Okay. Now moving to the next question, arrange the following in the correct developmental sequence related to microsporogenesis. So what is microsporogenesis? So in the anther there is a tissue which is known as sporogenous tissue, and this sporogenous tissue, one of the cell develops as a microspore mother cell. This microspore mother cell undergo meiotic division to form the microspores, and these microspores develop into the pollen grain. So this formation of microspore is known as microsporogenesis. So the sequence is very simple. First is the sporogenous tissue, okay, followed by the pollen mother cell BC, and then pollen mother cell will undergo meiosis to form the microspore tetrad BCA, and sorry, not B, sorry. First is the B, this is D, then A, then C. Okay, B, D, A, C. B is the sporogenous tissue, D is the pollen mother cell, A is the microspore tetrad, and C are the pollen grains. So B, D, A, C. So answer will be option number 3 here. Okay. Now moving to the next question. This question is from the molecular basis of inheritance, and here they are asking the steps of DNA fingerprinting. Now what is DNA fingerprinting? DNA fingerprinting is also known as DNA profiling. It is a method of basis of telling how the DNA in a population are different from one another. So how do we perform the DNA fingerprinting? Let's see step by step. First step will be the isolation of DNA. So from a cell, the DNA will be isolated, and after that, this DNA will be digested with the help of restriction endonuclease. So first A, okay. Second, after the restriction of the DNA fragments, different fragments will form. Then we have to separate the fragments on the basis of electrophoresis. So A will be followed by E, okay. A will be followed by E. So option 3 and option 4 are eliminated here. Okay. After this, we will uh, we will transfer these separated fragments into a synthetic membrane like nylon membrane. So followed by C. Okay. Then after that, there will be the hybridization using the labeled probe, that is B, and finally it will be the option number D, detection of hybridization. So A, E, C, B, D will be the sequence here. Answer will be option number 1, A, E, C, B, D. Now let's move to the next question. Exploring molecular, genetic and species level diversity for the products of economic importance. So this question is from the biodiversity and conservation. So if we want to know a product for economic importance, in which way? First is the molecular, genetic and species level diversity. This is representing the bioprospecting. This question have came in the NEET previous years also. So this question is repeated question hai ye. Okay. Now next one. Which of the following statements are true with respect to the sex determination in honeybee? As you know that honeybee is a social animal. It is a social animal, and in this animal there are three groups of insects. One is the queen, second are female workers, and the third are male drone male drone. Okay. Now we have to see the correct statements here. Choose the correct answer from the following option. So which statements are true here? First, an offspring formed from the union of sperm and an egg develop as female queen or worker. So first statement is true. When the sperm fertilize with the egg, this develops into a female. Either it will develop into queen or it will develop into a worker. So this statement is true here. Okay. Second, an unfertilized egg develops as a male by parthenogenesis. This statement is also true. So what is parthenogenesis actually? It is a development of the organism from an unfertilized egg. That is called parthenogenesis. So in both cases, what you will see, as the females are developing from a fertilized egg, so that is the reason queen and females are diploid, and the males are developing from unfertilized egg. That's why the males are haploid here. Okay. So next question is this: a male has half the number of chromosome than that of female. It is true. As we can see, queen and female workers are diploid and male drones are haploid. So that's why they have the half chromosomes. So male has half number of chromosomes. So this is true. Next, male produce sperms by meiosis? No. They produce sperm by mitosis. Sperms are produced by mitosis. So as you know that male drones are haploid, so the sperm which they will form, that will also be haploid. So the division will be mitosis here, not the meiosis. Okay. Next, honeybee have a haploid diploid sex determination system. It is correct. As you can see, haploid are the male, diploid are the female. That means diploid organisms develop into female and haploid organisms develop into male. So that's why this statement is correct. So here we will see A, B, C, E. Okay. A, B, C, E are correct here. Answer will be option number 4th. Now, moving to the next question. Identify the correct sequence of step in each cycle of PCR, that is polymerase chain reaction. And this question is from the applications of biotechnology. As this question is one of the very famous question of NEET. I will say like almost every year one question is coming from the PCR. Last year also one question came from the PCR this year. And now this year they have given a very simple question, steps of PCR. Okay. First step is the denaturation, in which the two DNA strands are separated by heating them at a high temperature up to 94 degrees Celsius. It is followed by annealing of primers and followed by the extension of primers using the Taq polymerase enzyme. Okay, so answer will be option number 1 here. Next, which of the following statements are correct with respect to DNA separation, isolation and visualization? So first of all, we have to find the correct statements here. First of all, the cutting of DNA is done by the molecular scissor. Yes, molecular scissors are also called the restriction enzymes. So this statement is correct. Second, the DNA fragments separate according to their size in agarose gel upon the electrophoresis. So the separation is based on the size only. So the larger fragments, they will move slowly and the smaller fragments will move very faster. So that's why statement 2 is correct here. The separated DNA fragments can be seen without staining when exposed to UV light. They are mentioning a term without staining. No, we have to use the stain here. And the stain that is used here is known as ETBR, that is ethidium bromide. Okay, so this statement is incorrect. The separated DNA fragments when stained with ethidium bromide can be seen in visible light. As you can see, options C and D are giving the answers of each other. Okay, they can be seen but not with the visible light, with the help of UV light. So this statement is also incorrect. So here statement A and B are correct. Answer will be option number 4th. Now moving to the next question, the main criteria used for 5 kingdom classification, which was given by R.H. Whittaker. It is also a previous year question. It have came in the NEET, R.H. Whittaker. So 5 kingdom classification, there are 5 kingdoms represented as Monera, Protista, Fungi, Plantae and Animalia. So what were the basis? First, cell structure, yes, cell structure. Second was body organization, but not the presence of flagellum. C is incorrect. Reproduction is correct and phylogenetic relationship is correct. So A, B, B and E. Answer will be option number 1. A, B, D, E only will be the answer here. Now moving to the next question. Which of the following is a triploid cell? इनमें से कौन सा triploid cell होता है? If you will see here, central cell contain two polar nuclei. It contains two polar nuclei. So the condition is N plus N. Okay, it is not triploid. Primary endosperm cell is formed by the triple fusion. When a male gamete fuse with the two polar nuclei, that is the secondary nucleus. So in this case, the primary endosperm cell which will be formed, it will be triploid in nature. Zygote is diploid in nature and the synergids are haploid in nature. So answer should be option number 2. Very easy question from sexual reproduction in flowering plants. Now moving to the next one. So this question is again from the molecular basis of inheritance, but they are saying, let's see. Which of the following statements are correct? So we have to find the correct statements here. First, histones are organized to form 8 molecules called histone octamer. So histone octamer, yes, they are forming these histones are arranged, okay? They are forming the histone octamer. This statement is true here. Second, histones are negatively charged basic protein. No, they are not negatively charged. Histones are positively charged proteins. So statement 2 is incorrect here. Next, histones are rich in basic amino acids, that is lysine and arginine. This statement is true. They both are positively charged amino acids. Next, the positively charged DNA is wrapped around the histone octamer to form a nucleosome. So what is incorrect here? If you see carefully, they are writing positively charged DNA. No, on the DNA, charge is negative. DNA charge is negative. So this will be incorrect. Next thing, the packaging of chromatin at higher level require additional set of protein called non-histone chromosomal protein. This statement is true here. So A, C and E are correct here. Answer will be option number. Okay. Now moving to the next question. Which of the following is in situ conservation method? So this question is from biodiversity and conservation. So you will see here that in situ conservation, what are in situ conservation? Where the animals and plants are conserved in their own natural habitats. Okay. So wherever they are growing or where they are found in their natural habitat, we are conserving them. So according to this, if you will see seed bank, botanical garden and wildlife safari parks, they all are the examples of ex situ conservation. So answer should be option number 1, that is sacred group. These are example of in situ conservation. Next, in the lac operon, the Z gene, which is also represented as lac Z gene, codes for. So this lac Z gene codes for the enzyme beta galactosidase. So answer should be option number 4, beta galactosidase, and this question is from molecular basis of inheritance. Now moving to the next one, this is based on the growth regulators and their functions or effects. Very simple. देखिए, first 2,4-D is an artificial example of auxin. Okay, it is a synthetic auxin 2,4-D and it is used as a herbicide. We use this as a herbicide. So A will be 3 here. So A is 3 is given in only one option, that is option number 3. So automatically you can eliminate the other 3. See next, GA3, that is gibberellic acid. It will be functioning for the in the brewing industry. Okay, then kinetin for the nutrient mobilization, and ABA, that is abscisic acid, for the stimulation of stomatal closure. So answer should be option number 3 here. Now, next one is again from the application of biotechnology. Arrange the following steps of somatic hybridization. So this is we can say steps in the somatic hybridization. Let's see. First one, in the first step, we have to find the correct sequence here. Correct sequence. So it will start like isolation of single cell from two different varieties of plant. Like from the two plants, we will isolate two different cells. Okay. After isolating the cell, you will digest their cell wall. You will digest their cell wall by using the enzymatic method. Okay, so it will be A. Next, after digesting a cell wall, you will get the protoplast. Okay, so then there will be the isolation of naked protoplast, that is DAB. Okay, then fusion of protoplast, that is C, and finally the growing of hybrid protoplast, that is E. So DABC will be the answer. Okay, this question is from the respiration in plants. Okay, it is not biomolecule, it is respiration in plants. So here we have to find the respiratory quotient of a biomolecule used for respiration. So for the respiration quotient, you will see that this is palmitin, which is a fatty acid. Fatty acid. For the respiration of fatty acid, the RQ value is less than 1. It is less than 1. So answer will be not less than 0.5, not between 1.25 and 2, not 1. It should be between 0.5 and 0.9. So RQ is around here 0.7. RQ is around here 0.7. So answer should be option number 2. Okay. So after this, I will call Shiny ma'am to continue with the botany topics. Thank you.
Thank you so much, sir. Now let's continue with the rest questions of botany. I hope the paper went well for all of you. And now let's discuss the rest questions left for the botany part. Okay. So the question is, which of the following statements is not true about the universal rules for binomial nomenclature? This question is from Living World, and we have to tell which is not true. Which is not true. Okay, so first statement says both the words in a biological name when handwritten are separately underlined or printed in italics. This is correct. Next, the specific epithet in the biological name starts with the small letter. This is also correct. The species name starts with the small letter. Then the first word in the biological name represents the specific epithet, while the second component denotes the genus name. This is incorrect. And the last one is biological names are generally Latin. So since they were asking not true, so option number third will be correct. Now let's move ahead to the next question. Next question is the enzymes required for carboxylation in Kelvin cycle is. This question is from photosynthesis in higher plants and the part is Kelvin cycle or the dark reaction. And the enzyme which is required for carboxylation is Rubisco enzyme which is present in the stroma. So option number 2 is correct. Next, which of the following formula is correct for floral formula for the family Solanaceae? Solanaceae is also known as potato family, and the question came from the chapter Morphology in Flowering Plants. So for the Solanaceae family, the correct formula is option number 1, where we see actinomorphic, bisexual, calyx fused or gamosepalous condition. Then petals also fused and 5, gamopetalous condition. Androecium 5, hypogynous ovary. Okay. And also epipetalous condition is C. So option number 1 is correct. Next, which of the following types of pollination brings genetically different types of pollen grains to the stigma? So this question is from the chapter Sexual Reproduction in Flowering Plants and the part is pollination, and they're talking about which type of pollination is genetically different. Genetically different is the xenogamy, which is also called as cross pollination. So option number 3 is correct. Now let's move ahead to the next question. Next question is a match the following type of question from the chapter respiration, where we talk about the four processes of that occurs in the respiration process. First is glycolysis. Glycolysis occurs in the cytoplasm. Then next is ETS. It occurs on the inner mitochondrial membrane. Then we have accumulation of protons that occur in intermembrane space. And the last is Krebs cycle, which occurs in the mitochondrial matrix. So the correct option is 1, uh, 3. A3 is only for option number 2. So correct option is option number 2. Now let's go ahead. Next question is the mutant codon of beta gene causing polymerization of hemoglobin and change in RBC shape is. This question is from molecular basis of inheritance and it talks about the mutation. The correct option is your GUG, where GUG. Okay.
Next is, which of the following is an appropriate example of sexual deceit? Sexual deceit, my dear students, we have studied in the chapter organism and population. Okay, and there we have studied for the certain examples, and the one of such example of sexual deceit is seen in Ophiris or orchids and bumblebee, where the female bumblebee, it tries to act or show as a flower, the flower, it represents the female bumblebee, so the male comes and gets it pollinated. Now the next question is, in which of the following ovules are not enclosed by a ovary wall and remains exposed? The question is from the chapter Plant Kingdom. The options are Flagelella, Funaria, Pinus, Wolfia. So the which is not enclosed by ovary wall and remain exposed. So they are asking about exposed. So the correct answer will be Pinus, which is a gymnosperm. Next, what is the probability of having children with O blood group where both mother and father are heterozygous, and for A and B blood group respectively? This question, dear students, is from the genetics or the chapter principle of inheritance. Here they are saying that the mother's blood group is IAI or A and father's is B, and they are heterozygous. So when they are crossed, what is the probability of getting a child with O blood group? For O blood group, it should be small I, small I. So we can simply perform the cross. So with this cross, you can know that the probability of getting small I together or getting O blood group is 25%. Hence the answer will be option number. Next question is non-membrane bound cell organelles found in both prokaryotic and eukaryotic cell. The question is from the chapter Cell, the Unit of Life, and here they are asking which is the non-membrane organelle which is present in both eukaryotes and prokaryotes, and the answer will be ribosomes. Next question. Ecological pyramids represents the relationship between the organisms at different trophic levels, and they are generally inverted form. The question is from the chapter ecosystem, and we see that inverted pyramid is observed in pyramid of biomass in sea. Moving on to the next question. The next question is also a matrix match question from the chapter Microbes in Human Welfare. Here, streptokinase is your removal of clots in blood vessels. Then we have statins, blood cholesterol lowering agent. After that, we have lipases, detergent formulations, and cyclosporin A, which is an immunosuppressive agent. So, according to this, the correct option is A2, B3, C4. A2, B3, C4, option number first is correct. Now, let's move ahead to the next question. Now, the next question is, choose the correct statements regarding cell organelles and their inclusion. This question is also from the chapter Cell, the Unit of Life, and they're asking us to find the correct statements. The first is endomembrane system includes Golgi complex, endoplasmic reticulum, and mitochondria. This is wrong. Second, rough endoplasmic reticulum bears ribosomes on its surface. This is correct. Next, both mitochondria and plastids have circular DNA. This is also correct. A network of microtubules, microfilaments, and intermediary filaments present in the cytoplasm is called cytoskeleton. This is also correct. Now moving on to option number E, mitochondria, a single membrane bound structure. This is wrong. So since they are asking us correct answer from the given, correct statements from these given statements, the option B, C and D is correct, which is option number 4. Next question. In a population of a grasshopper species, the chromosome number of some members is 23 and some of the members possesses 24 chromosomes. The 23 and 24 chromosome bearing members in this species are. This question is from the chapter Principle of Inheritance, as you can see, and from the topic sex determination. Here we see that in grasshopper, XX XO type of sex determination is seen, where XX represents the female and XO the male. So here the 2n is equals to 24, which represents female, and 2n minus 1, which is 24 minus 1, that is 23, which represents the male. So the correct answer will be, they are asking 23 and 24, means 23 is male and 24 is female. So the option will be male and female respectively, which is fourth option. Next question, in which animal do haploid cells divide mitotically to produce gametes? This question is from the chapter Cell Cycle and Cell Division, topic mitosis. Here we see that haploid cells undergoes mitosis in the case of male honeybees, which are also called as drones. Moving on to the next question, cell the unit of life. The question comes as, select the correct statements regarding cell membrane in eukaryotic cell. Statement 1 says the membrane of human RBCs has approximately 52% protein, which is correct. Next, major phospholipids are arranged in a bilayer. Statement B is also correct. Statement 3, extension of the plasma membrane into the cell forms mesosome. This is also correct. And tail towards the inferior part of lipids are hydrophobic and thus protected from aqueous medium. This is also correct. Next is glycocalyx is present on the outer surface of the plasma membrane. This is incorrect. It is present on the outer surface of the cell wall. So the correct option will be option number 1, 2A, B. What is that hydrophobic from? So option will be A correct, B correct, and C, the correct option is fourth one. Most appropriately correct option is fourth one, A, B, D only. Next, which of the following equation depicts Wellhurst-Pearl logistic population growth? The question is from the chapter Organism and Population, and here they have given direct equation for this Wellhurst population or exponential growth, which is option number 1, dn by dt is equals to Rn k minus n by k, where this k minus n by k represents your environmental resistance. I hope the questions are clear. Now moving on to the next question. What is the reason behind production of large holes in Swiss cheese? The question comes from the chapter Microbes in Human Welfare, and the options are as follows. The production of large amount of CO2 by Clostridium botulinum, this is wrong. Next, the production of large amount of carbon dioxide and hydrogen by Trichoderma polysporum, which is also wrong. The production of large amount of carbon dioxide and hydrogen by lactic acid bacteria called lactobacillus, this is also wrong. Next is the production of large amount of carbon dioxide by propionine bacteria Shermini, this is correct. So the option number 4 is correct. I hope you understood all the botany questions. Now moving on to the zoology discussion, I would like to invite Mahima ma'am for further discussion of the questions.
Hello students. Thank you ma'am for introducing. Hello students. So I hope you all did very well. The paper was very easy. Okay, it was moderate. I think you have did very well. Now let us quickly, I think we are done with the botany discussion. Let us quickly start with the zoology paper discussion. Okay, coming on to the first question students. Okay, now let us see the first question. Yeah. Okay, so identify the correct statement. So if you see, they are asking in this question to identify the statement which is correct. So identify the correct statement.
Yeah, identify the correct statement about the biomolecules. So let us see the first statement. Okay, so lipids are generally water soluble. Okay, they are not water soluble, they are water insoluble. Okay, so first statement is incorrect. Next, proteins are polypeptides, right? Okay, NCRT lines again. Okay, so they are polypeptides, that's right. Polysaccharides are long chain of sugars, right? They are the long chain of sugars. Now, adenine and guanine are substituted pyrimidines. No, they are not substituted pyrimidines. They are purines. Okay, this is wrong. Now, almost all enzymes are protein. Yes, so they almost all the enzymes are protein. So that statement is also what? Correct. So as in the question they are asking correct statement. So what exactly will be? It will be B, it will be the C, and it will be the E option. So here the option number 2, B, C and E will be the correct one. Okay, let us go to the next question. Now, which of the following statement, again which of the following statement are not true? Okay, what is it they are saying? They are not true. So we should check for the incorrect one. Now, which of the following statements are not true regarding the restriction endonuclease? Now, again this question is taken from the chapter Biotechnology, Principles and Process. Now, they are called molecular scissors. That's right. Okay, not true. So, it's false. Now, these enzymes are responsible for restricting the growth of bacteriophage in the E. coli. Absolutely. Okay, so that is how they were discovered. Fine. Now, next, they cut the DNA only at the center of the palindromic sequence. Do they cut at the center? Exactly at the center? No. Okay, they read up the sequence, okay, and at the specific site they do. They don't directly cut it at the center. So, this statement would be incorrect. Right? Next, they remove the nucleotides only from the end of the DNA fragments. Students, now you should be kind of, have a look here properly, because in the question they are asking what? Restriction endonuclease, and they are saying in the statement that these restriction endonuclease remove it. Okay, so from the end. Now, how a restriction endonuclease could remove the nucleotide sequence from the end? So, this option gonna be incorrect. Okay, now next they recognize specific palindromic base pair sequence. Absolutely right. Okay, this is absolutely right. Now again in the question they are asking false, which is not correct. So what would be it? It will be the option number C and D. So we can go with the option number 3. Okay, I hope you have marked it correctly. Let us go for the next question. The very easy question, directly aligned from the NCRT. Okay, they are asking alpha helix is found in which level of protein structure? Now this question is taken from what? Biomolecules. Now when we know that we have the primary structure, secondary structure and tertiary structure. So primary is like a chain, okay, or straight chain. When we talk about the secondary structure, you find two forms. Okay, you form the helix form, you form the beta pleated one. Now in the diagram, NCH diagram, they have clearly mentioned it. Now as we are talking about clearly the alpha helix, for no doubt the answer gonna be secondary. The answer gonna be secondary structure, so option number 4. Okay students, I hope you have marked it correctly. Let us go for the next question, which is a matrix matching. This question is taken from the chapter what? The biomolecules. Fine, and a few of the application part. Now I hope, let us see how exactly it is. Now they're talking about genetically modified. So we know that the genetically modified is nothing but the Bt, cotton, bacillus, thuringiensis. Next, thermostable DNA. Now how do we make this thermostable DNA? We use what? The thermus aquaticus. Then Ti plasmid. What is a Ti plasmid, students? When we talk about the Ti plasmid, we talk about the Agrobacterium tumeniferans. Okay. Now next we have the PPR322, which is nothing but the Assertia coli. So here the genetically modified. So option A should be 1, A should be 2. Okay, let us see. Yes. Next B should be what? B should be 3. C should be 1 and D should be 4. So option number 3 is the correct one. Okay. Next, let us go to the next question. Yes, fine. So this question is also from the chapter Biotechnology and its application. Let us quickly read the question. Now, exploring molecular, genetic, species level diversity for product and economic importance. What exactly this is called? Okay, let us see the option. It is not biomagnification. In biomagnification, there would be accumulation there of the toxic substance. So we can eliminate this option. It is not biofortification. It is not the bioremediation as well. Okay, but what exactly it is? The bioprospecting. Again, a direct line from the NCRT students. Okay, next, let us move on to the next question. Now here in this question is taken from the chapter Biotechnology Principles and Process. Now here they are talking about the PCR, and in the question they are asking that what are the sequence? Okay, the most easy question. Now let us, and we need to look at for what is the proper rearrangement then. Okay. Now identify the correct sequence and steps in each cycle of the PCR. So we know that it consists of 3 steps which include what? It includes denaturing, annealing and what? Extension. Fine. So here which option is correct? So the denaturing, annealing, the extension, the very first option if you see, okay, it is the one. So very simple, right? And previously also many times it has been asked in the exam. Next, let us move on to the next question, students. Now, this is statement based question. Now, what exactly they are asking in this question? Now, which of the following statements are correct with respect to DNA separation, isolation and visualization? So, here we are talking about a process called now this gel electrophoresis, and this question is from the chapter Biotechnology Principles and Process. Okay, so let us go through the statements. Now here they are saying the cutting of DNA is done by molecular scissors. Yes, now molecular scissors is another name for what? The restriction enzymes. Okay, next the DNA fragments separate according to the size in an agarose gel upon the electrophoresis. Absolutely right. Okay, so they are segregated upon according to what? The size there. The separated DNA fragments can be seen, they are saying without. So students, do you think it's not without? They can be only visualized when we stain them using the dye, that is etherium bromide. So this statement, now they are saying the separation of DNA fragments can be seen without staining when exposed to the UV light. Okay, so what is incorrect part? They are saying without, but we know that it is with the staining. Okay, so this statement would be wrong. Next, the separated DNA fragments when stained with etherium bromide can be seen in the visible light. Okay, can we see them in the visible light? No, we can only see them under the UV rays. Okay, so where we can see them? It is what? The UV rays. So this statement is also incorrect. Now what they are asking in the question, okay, what they are asking in the question, they are asking the correct. So which are correct? Okay, the first and the second, that is the A and B statement only. So your option number 4. Okay, let us move on to the next question. Okay, so this question is taken from the chapter Evolution. Fine. Now again, if you have read the NCRT thoroughly, you know that this is a direct NCRT line. Okay, let us quickly go through the question. Now, since the origin and diversification of life on Earth, there have been 5 episodes of mass extinction. Now, how is the 6th extinction, which is in progress, different from the previous episodes? Okay, so the current species extinction rates are far lower. Is it far lower? No, it's more high. Okay, so this statement is incorrect. Far lower than those in the previous episodes. The present species extinction rate is 100 to 1000 times faster than in the pre-human times. That's right. Okay, the present net species extinction rate is 0. Okay, it's not 0. What do you mean by 0? Like for example, the number of new species which are, formed and the one which are extinct. Okay, the both ratios should be same. Now, is it same? No, it's not same. Okay, so this statement is what? Incorrect. Next, the current species extinction rate is nearly 10 times faster than the previous episode. Okay, so here they are saying 10 times, but it's not 10 times in real. It's somewhere in the range between 100 to 1000. So this statement is what? Incorrect. Now in the question they are asking which statement, okay, what is the correct one? Fine. Now which is now it will be what? The presence of species extinction, which is from 100 to 1000 range. Fine students, now let us move on to the next question. Okay, again we have a matrix matching. Now here the trypsin. Now we know that what exactly is a trypsin. We know that trypsin is an enzyme. Okay, what is the morphine? We know morphine is what? It is an alkaloid. Now what is in concavenin? It is a lectin. And what is collagen? It's an intercellular ground substance. So what would be the correct option? Option A should be 3. Okay, option B should be 4. Option C should be 2 and option D should be 1. So we can go with the option number 1 without any doubt. Okay, again a very easy question. Now let us move on to the next question, students. Now insertion. Okay, this question is taken from the chapter again, biotechnology principles. Now let us go through the question. Insertion of a foreign DNA at BAM-H1 site in an E. coli cloning vector PBR322. So when we talk about PBR322, so we have two sites, right? So here we have an ampicillin, okay, and here we have the tetracycline. Now here the site which is there is what? The BAM-H1. Now here what we have PVU1 and then we have PST1. Yes or no? So let us read in the question what they are saying. So insertion of a foreign DNA happened. Where it happened? It happened at this site, okay, at the BAM-H1 site. Fine. Now, in an E. coli cloning vector, it results in the loss of an antibiotic resistance. So, obviously, when it is, inserted at that place, okay, what exactly happened? The resistance for tetracycline would be lost. Okay. So, the resistance for tetracycline will be what? Lost. It is not for the ampicillin and not for the other. We can quickly eliminate the options. It's a very easy question. I hope you all have marked it correctly. So the answer for this question will be the option number 3. Now let us move on to the next question. Okay. Now again, this is from the human reproductive health. Okay. So the correct statement regarding the gift to overcome infertility. So we know that this is from the ART, the assistive reproductive technologies, right? So here they are talking about what? The gift. Now we need to find the correct statement which is regarding this gift there. Okay, so ova collected from a female donor are transferred to the uterus. Okay, so we know that what happens here. Now the female is unable to produce a ova, so it is taken up from what? The donor. So again, do we place it in the uterus? No, right? It's in the fallopian tubule. So this statement is what? And they're saying infertile female. So this is absolutely wrong statement. Next, it is a transfer of an ovum collected from a donor into the fallopian tube of another female who cannot produce ovum but can provide suitable environment. Okay, but can produce suitable environment for fertilization and development. Okay, right. Fine. Let us still go on to the next two statements. Early embryos up to 8 blastome years are transferred into the uterus. Okay. Do we transfer them? Okay. Now, here they are talking about ZIFT, right? In an infertile female. Next, early embryos with up to 8 blastome years are transferred into the fallopian tube of an infertile female. They are saying infertile female. Okay, so infertile female may it's now the early embryos up to 8 blastome years are transferred into the fallopian tube into an infertile female. So when we see technically this would be what the correct statement. Now next. Yes. Now, evolution of human appears parallel to the progressive development of the brain and the language, and such as the evolution of individual species in the sequence. Now, we know the sequence, which is the most important topic now from the evolution chapter, right? So, we know it is Dryopithecus followed by Ramapithecus. Now, let us see. If you see these options, you can eliminate the remaining options by that, because first is Dryopithecus followed by Ramapithecus, right? So here you can eliminate the remaining three options easily, and then Ramapithecus, then Homo habilis, Homo erectus, Neanderthal, and what? The Homo sapiens. So the second option will be correct for this. Next. Okay, the fetus movement and hair appears on the head. Okay, in where the 20 weeks of the pregnancy. Now, fetus develops limbs and digits. That is 8 weeks of pregnancy. Fetus develop external, genital organs. That is 12 weeks of pregnancy. Fine. Now, fetus body is covered with a fine hair. Eyelids and eyelashes are separated. Okay, that are formed in 24 weeks of what? The pregnancy. Now, here if we see this, the option number 4 will be right. Next. Now, a group of researchers procured some fish like animals, and upon investigation, the following characters are there, and we need to select the option which is right. Which species does they belong to? Now, when we see the first one, endoskeleton is made up of cartilage. Okay, it is an ectoparasite. Now, when you see the word ectoparasite, it is clear the thing that comes in your mind. Okay, you can easily guess the option, but still, let us see the another one. Okay, ectoparasite, as they were found attached to the fish skin with the circular sucking mouth, they're like circular, that is suctorial. You know it's cyclostomes, right? Next, Paired fins, scales were absent, but 7 pair of gill slits were present. Now, when we see this option, okay, what will be the correct one? The petromyzin. It can't be the exocetus. Fine, it can't be the brachystoma. Fine, it cannot be the scoliodon. So, directly you can mark the second option. That is what the petromyzin. Fine. Now, next, let us go to the next question. Now, this question is from the human reproduction. Okay, so here they are talking about the process called spermatogenesis. Now let us see, spermatogonia undergo a series of cell division to produce sperms. Now they are asking us to select the correct statement. What statement we need to select here? The correct statement. Okay, from the following. Spermatogonia always undergo meiotic cell division. Okay, so they are saying spermatogonia, they always, and they are asking us to correct the correct one. So this is what the wrong. Now primary spermatocytes, let's see what exactly happens. Some of them they will differentiate, okay, and only they will be going into the next part. Fine. Now primary spermatocytes divide mitotically to produce secondary spermatocytes. It is meiotically given here, it's wrong. Next, secondary spermatocytes through the second meiotic division produces haploid spermatids, okay. Next, spermatids produce spermatozoa through mitosis. Okay, it's not. Next, spermatids transform into spermatozoa by spermiogenesis. Okay, that statement is what? Correct. So we can go with the option number 1, that is C and E are the correct one. Fine students, so I hope you have marked all the questions properly. Now the next set of questions will be discussed by Kavya ma'am. So I would like to call Kavya ma'am to continue the discussion for the next set of questions then.
So let us go to the next set of questions from the zoology part. The paper was easy, right? The bio paper was damn easy. Yes, I agree it was a little lengthy paper, but the paper was very easy. And most of the questions were though the statement type of questions, right? Okay, so let us move to this set of questions. So coming to the first question, arrange the following events occurring in the renin-angiotensin mechanism in the correct order. Okay, so we are asked about the RAS mechanism, and it is a question which is being taken from the excretory products and its elimination, though human physiology has the greatest weightage, right? So if you see over here, first RAS mechanism, where does it start? The RAS mechanism starts whenever there is a fall in the GFR. See, in all the statements, where is the fall in the GFR given? The fall in GFR is given over here, right? So this is what is exactly the first one. So it will be starting with C. So automatically the two options are being eliminated. The automatically two options are being eliminated. Let us see. After there is a fall in an GFR, what exactly happens? Obviously, the renin enzyme is released. That renin enzyme is activating the angiotensin 1. Angiotensinogen is converted into angiotensin 1, and angiotensin 1 is further converted in the presence of the same enzyme into angiotensin 2. So what will be our second one? This will be the second statement. Then after the angiotensin 2 is activated, what happens? What is the function of angiotensin 2? Angiotensin 2 is responsible for vasoconstriction, and because of vasoconstriction there will be release of the aldosterone. So this is statement number 3. Next, what will happen? Because of the release of aldosterone, what exactly happens? There is reabsorption of the sodium ions and the water molecules. There is reabsorption of sodium ions and water molecules. So this is number 4. And last but not the least, because of their reabsorption of the sodium and the water molecules, automatically the blood pressure will be increased, and thus the GFR will also be increased. So what is our order if you see? C, E, D, B, A. So our correct option is option 4. Moving on to the next question. This is a matching matrix types of question which have been taken from the chapter breathing and exchange of gases. This is from the subtopic pulmonary volumes and capacities. But this is the easiest question that could be given from that chapter. You should just know the volumes and capacities
thoroughly, right? Okay, so if you see here, they are asking you in the list 1 they have given respiratory volumes, and in the list 2 they have given you the capacities. So if you see expiratory reserve volume is 1000 to 1100 ml. So this is 3. Next, residual volume. The residual volume is 1100 to 1200 ml. Next one is inspiratory reserve volume. Inspiratory reserve volume is 2500 to 3000 ml. So it will be option 1. It will be stated as number 1 from the list 2. Tidal volume is 500 ml. So it is 2. So 3, 4, 1 and 2. Which option is matching? 3, 4, 1 and 2. Option 3. Moving on to the next type of question. The next type of question is again a matrix type of question which has been taken from the chapter reproductive health. So from the reproductive health, this is about the barrier methods. This is just nothing but the contraceptive methods, the birth control methods. So let us see. So here it is given in list A, they have given certain contraceptive methods and they have just defined what is exactly it in list 2. So if you see here, progestasert, we know it is a hormone releasing IUD. Multilode 375 is a copper releasing IUD. Diaphragm is just diaphragm is nothing but a barrier method. Diaphragm is nothing but a barrier method. And coming to Saheli, we know it's once in a week pill, but that is nothing but a oral contraceptive. So 3, 4, 1 and 2. Option 1. Moving on to the next question. This question is taken from Animal Kingdom. The question is, the flightless bird with 4 limbs modified as a paddle-like structures suited for swimming is known as. First, let us eliminate the flight birds. Okay, that will make the question a little easier. So in this, Psittacula is a flight bird, so it is eliminated. Neophron is a flight bird, so it is also eliminated. What are left? Shrewdio and Aptenodytes. So if you see Shrewdio, in case of Shrewdio, is the flight limbs modified into certain case? No, the flight limbs or the 4 limbs are not modified. But if you see Aptenodytes, that is nothing but penguin, the 4 limbs is being modified into paddle-like structures for the purpose of swimming. So it's option 4. Moving on to the next set of question. This question is from the chapter Animal Kingdom. In this question, they have asked, which set of fish is belonging to the class Osteichthyes? It is belonging to the class Osteichthyes. Let us see the first option, devilfish, cuttlefish, hagfish. Devilfish, cuttlefish, both of them are mollusk, whereas hagfish is a cyclostomata. So this question is eliminated. Moving on to second, starfish. First itself, starfish is an echinoderm, so it is not the option. This is also eliminated. Flying fish, angelfish and fighting fish. Yes, these all 3 of them are belonging to the class Osteichthyes. Moving to the next question. This question is a little mathematical question which is being taken from the chapter Body Fluids and its circulation. So if you know the entire flow chart of the components of blood, this question is very easy for you. So the WBC count of a person's blood sample is 8,000. How many eosinophils and lymphocytes would be in the same blood sample approximately? So first you should be knowing actually the percentages. If you know the percentages, there itself you can solve for the number. So eosinophils, what do you know? 2 to 3 percentage, whereas lymphocytes is 20 to 25 percentage. Now let us solve for 2. For the minimum count, 2 percentage of 8,000 is nothing but 160. Next, for the maximum count, 3, right? So 3 percentage of 8,000. Okay, 3 percentage of 8,000 is nothing but 240, right? It is nothing but 240. So the count for eosinophils will be 160 to 240. Will be 160 to 240. Moving on to the lymphocytes, 20 to 25 percentage. So 20% of 20% of how much again? 8,000, right? So what will be the count? The count will be approximately 16,000, whereas 25% of 8,000 is nothing but 2,000. So the count will be 1600 to 2,000. So which option will be matching? Option number 1. Moving on to the next question. This question is from biotechnology and its application. The toxin proteins isolated from Bacillus thuringiensis coded by which of the following genes would control the cotton bollworms and corn borer? This is a direct question. So cotton bollworms, it is from actually cry1 AC, and corn borer is cry1 AB. So third option is the most appropriate option. Moving on to the next, it is again a matrix type of question taken from the chapter Human Health and Diseases. It is from the subtopic of drugs. Okay, so if you see nicotine, so nicotine, what is nicotine doing? Nicotine is stimulating the adrenal gland to release catecholamines into blood circulation, which will increase the heart rate, and simultaneously it will also increase the blood pressure. So nicotine matches with number 2. Morphine. Morphine is actually extracted from the poppy plant. It is extracted from the poppy plant, which is acting as a sedative painkiller. It is acting as a sedative painkiller. Next one, heroin. Heroin is also acting upon the central nervous system and gastrointestinal tract. It is acting as a depressant, which is slowing down the body functions. 4. Cocaine. It is causing the sense of euphoria and increased energy because the receptors are directly attached to the central nervous system. So 2, 3, 4 and 1. Second option. Moving on to the next, again a matrix type of question from the muscular disorders, musculoskeletal disorders from the chapter Locomotion and Movement. Okay, we'll try to solve it faster. Tetany, we know tetany is because of the low calcium level in our body. So because of the low calcium, there will be muscle spasm. So here, wild contraction of muscle due to low calcium in the body fluid. Arthritis is simply, it is because of the inflammation of the joints. Do you find anywhere 3 and 1? Yes, option 1. 3 and 1. Okay, next. This question is taken from the chapter Breathing and Exchange of Gases. We have a sector in our NCRT textbook which is giving the series of the respiration, and exactly that series is given in this question if you have read the NCRT thoroughly. Okay, in humans, respiration occurs in the following steps. Arrange these steps in the correct order. So first and foremost, what usually happens, it is the pulmonary ventilation by which the atmospheric air is drawn and CO2 rich alveolar air is obviously released out. So the first statement is pulmonary ventilation. As soon as the air is entering, what will happen? It will be exchanged. Where is it getting exchanged? It is exchanged at the alveolar membrane. Now, after being exchanged at alveolar membrane, who will transport it? It is transported by the blood. The gases, the respiratory gases are being transported by the blood. They are being transported by blood. Now what happens? They are reaching the tissues. In tissues, again, they have to give the oxygen that it has brought, right? So there will be exchange of the respiratory gases in the tissues, exchange of the respiratory gases in the tissues. And last but not the least, in the cell, what will happen? Oxidation of glucose, that is nothing but the cellular respiration. That is nothing but cellular respiration. So C, B, and then we have E. C, B, E, A, and D, right? Moving on to the next question. Arrange the following cell layer structures around the female gamete from outer to inner side. Such words are very important in terms of exam because so you are supposed to arrange them from outer to the inner side. This question is from human reproduction. So if you see, they have given zona pellucida, corona radiata, perivitelline space, and the plasma membrane of the ovum. So corona radiata, we know it already that it is the outermost layer. And zona pellucida, when we are talking about zona pellucida is nothing but a glycoproteinaceous layer which is produced by the ovum itself, right? So this will be the first one, this will be the second one. Peripetalene space, it is a fluid filled space which is present between the zona pellucida and the plasma membrane of the ovum Last we have the plasma membrane of the ovum. So from outer it should be C, A, T and D. Right? Moving on to the next question. The human protein alpha-1 antitrypsin obtained from transgenic animals is used for the treatment of, directly from NCRT. Wherever you hear the term alpha-1 antitrypsin, it is for the treatment of the emphysema. It is nothing but a respiratory disorder. Moving on to the next question. Male frogs can be distinguished from female frogs due to the presence of. This question is taken from the chapter Structural Organization in Animals from the topic of frog. So you should be knowing the difference between male and female frog. We have only two differences. What are the two differences? One is vocal sacs and the other one is copulatory pads. Vocal sacs, the male have the vocal sac so that it can call the female, attract the female during the breeding season. Copulating pads are present on the first digit of the forelimb, which help in attachment or grabbing the female during the amplexus or copulation. So if you see, are those two options there? Yes, copulatory pads is there and vocal sacs is also there. Option B and D. So I hope these set of questions you must have solved correctly. Now the next set of questions from zoology will be explained by Bhavna ma'am.
A very good evening to one and all. And this is Dr. Bhavna, Zoology faculty. Let us go into the next set of questions. Hope you all have done well in the NEET exam. Biology paper is very easy. Okay, let us continue with the next set of questions. Yes. Now, the first question, choose the correct statement regarding the frog's anatomy. So, hepatic portal system is the special venous connection between the liver and the intestine. And then we have, there are 12 pairs of cranial nerves arising from the brain. And then the ureter and oviduct open separately into cloaca in female frogs. Hindbrain consists of cerebellum, medulla oblongata and optic lobes. And sinus venusus join the right atrium of the heart. Here they are asking choose the correct statements. And this question is from structural organization in animals and mainly it is about frog. So which are the correct statement? The first statement is correct and the second statement there are 12 pairs of cranial nerves is wrong. In frog there are only 10 pairs of cranial nerves and then the ureters and oviducts open separately. This is correct. Hindbrain consists of cerebellum, medulla oblongata and optic lobes. Anytime hindbrain consists of only cerebellum, medulla oblongata and pons. It is not optic lobes. Optic lobes belongs to that of midbrain. So that is wrong. And then sinus venous joins the right atrium of the heart. So as they asked to choose the correct statements, A, C and E are correct. Okay, let us go to the next question. Yes. Which of the following statements are correct with reference to human endoskeleton? This question is from locomotion and movement. Human skull is monocondylic. So human skull is always dicondylic. It is not monocondylic. Next, the joint between any two adjoining vertebrae is called cartilaginous joint. Yes, between two vertebrae, definitely there is cartilaginous joint which is present. Fibrous joint is present associated with cranial brains in the skull. So this statement is correct. In humans, the number of cervical vertebrae is 7. Yes, the number of cervical vertebrae is 7. 7 cervical vertebrae, 12 thoracic and lumbar vertebrae and we have coccygeal and sacral vertebrae. But here the statement is correct. All the ribs except the last pairs are bicephalic. This statement is wrong because all the ribs are bicephalic only because they all have two heads. So two heads means here backside they are attached to the vertebral column. So this statement is wrong. The occipital bone of the skull is articulated with atlas vertebrae. So backside there is occipital bone and it is articulating with that of the first cervical vertebrae, which is atlas. So choose the correct answer from these is we have only like B, C and E. So the option is third option. Next question. Match the following type of question, and this question is from chemical control and coordination. In this chemical control, directly match, they have given the hormones and what is their function they have given. Suppose cortisol. What is the function of cortisol is it produces anti-inflammatory reactions. Anti-inflammatory, suppress the immune response and all is done by the cortisol. When we come to aldosterone, aldosterone basically stimulates the reabsorption. Anything reabsorption of sodium, water is done by aldosterone. And then we have cholecystokinin, which is a gastric enzyme. It will stimulate the secretion of pancreatic juice and then bile juice. And then last we have progesterone. Progesterone directly it will help in stimulate the formation of alveoli in mammary glands. So if we see the option, it is 2, 3, 4 and 1. Directly we can go with the second option. Next question. The following are the stages of life cycle of Plasmodium. Arrange the stages in the proper order. So this question is from human health and diseases, and this is the life cycle of malaria. So first when we start with this, the female mosquito injects sporozoites into the humans during the bite. First is as soon as the mosquito will bite, immediately the sporozoites will enter into a healthy female or a healthy human. Then second, the sporozoites will reach the liver through the blood they reach. And third, it will go into again liver cells. First liver cells are affected and then the RBCs are affected. So liver cells and finally it is RBC and the last we have is the gametocytes which develop in the RBCs. So it is like a sequence. They ask the question as arrange it as sequence. So the sequence is EDBAC. Second option. Okay, next question. Select the incorrect statements from the following. And this question is from animal kingdom. Digestive system in platyhelminthes is incomplete. Yeah, only one opening is present for both mouth and anus, so it is incomplete. Bilateral symmetry is a characteristic feature of adult echinodermata. This is wrong. Why? Because bilateral symmetry is a characteristic feature is wrong because radial symmetry is there for adult echinodermata. Pseudocoelom is possessed by Ace clementhus. Yeah, this is correct. We have acoelomates, pseudocoelomates, and coelomates. So under pseudocoelomates, we have Ace clementhus. Notochord is persistent throughout the life in the class Chondrichthyes. Notochord is there compulsory in the Chondrichthyes group because they are belonging to cartilaginous fishes and notochord is persistent. Next, members of class Reptilia maintain constant body temperature. Reptiles, they don't maintain constant body temperature. They are not apes and mammals maintain constant body temperature, whereas reptiles, they are basically poikilotherms. Poikilotherms means they will change wherever they go. So they are asking select the incorrect statement. So which is incorrect is B and E are incorrect. Next question. The specific receptors for neurotransmitters in a synapse are present on. So whenever there is a synapse means it is a junction between one neuron and another. So definitely the receptors will be present on postsynaptic membrane. From presynaptic membrane to postsynaptic membrane, the impulses are transferred. So where are the receptors present means it is directly the postsynaptic membrane. And next question. Choose the correct statements regarding the muscle contraction. So in this question is from directly locomotion and movement mechanism of muscle contraction. And here we have a motor neuron carries a signal sent by CNS to the sarcolemma of the muscle fiber. Yes, from the neuron first a muscle should get an impulse. So that is correct. Then the neural signal generates an action potential which causes the release of calcium. Calcium is immediately released and there will be definitely increase in calcium levels. But increase in calcium inactivates actin is wrong because here for increase in calcium causes the cross bridge formation. So this is wrong. It is given here as breaking of cross bridge. Actually they should give you as formation of cross bridge. So this statement is wrong.
Actin binds to myosin head to form a cross bridge. This is correct. Actin and myosin bind together. And shortening of sarcomere take place by pulling actin filaments towards the center of A band. So here I band will reduce, A band will retain its length, Z lines all will reduce. So here the pulling of actin filaments and all is done. All are correct statements only except C. So we go with the first option. Which of the following is not an example of convergent evolution? This is a direct question from evolution chapter, and these is not an example of convergent evolution means they are asking about homologous organs. Homologous organs have same origin and all, and they exhibit divergent evolution. Direct question is divergent evolution is exhibited in forelimbs of whales and bats. Rest all, if you see either eyes of octopus and mammals or wings of butterfly flippers, all these have analogous organs. Only this one have homologous organs which have same origin. Next question. The JGA is a special sensitive region formed by cellular modifications of. This question is from excretory products and elimination. What is JGA means? Juxtaglomerular apparatus. It is formed by a combination between the DCT and efferent arteriole. Efferent arteriole it is. DCT cells are called as macula densa cells and efferent arteriole cells are called JG cells. So directly we can go with the third option. DCT plus efferent arteriole forms the JGA. Next question is from biomolecules. The following reaction depicts the activity of a particular class of enzymes. So this is in enzymes classification it is given, and here we have directly it is lyases. Why we see here the lyases is because it will help in even removal of groups other than hydrolysis. If they give you hydrolysis group then it is hydrolases, but other than hydrolysis it is lyases. Next question is directly again from animal kingdom. We have matched the following. Here they are asking which type of respiration is present. Suppose if you see mollusca group, they have gills. So definitely it is the branchial respiration. And reptiles means they live on land, snakes and all. They exhibit pulmonary type of respiration. Now adult amphibians exhibit both the pulmonary respiration as well as cutaneous respiration. Both of the respirations they exhibit because amphibians are living on land and water. So both types of respiration is exhibited by them. Next we have amoeba. Amoeba exhibits cellular type of respiration because it's by simple process of diffusion. So directly we can go with 2, 1, 4 and 3. Second option. Okay, next question. This is a question from directly evolution. Here they have given you years and they asked you to match which year what event has occurred. Suppose 65 million years ago, then there are dinosaurs that disappeared from the earth. Suddenly dinosaurs have disappeared. Then we have 500 million years ago, invertebrates are formed. Direct memory based question it is. And 350 million years ago, there are jawless fishes, and 320 million years ago it is dinosaur, and it is basically the seaweeds which are there, seaweeds and few plants. So directly we can go with the option as 2, 4, 1 and 3. 2, 4, 1 and 3. This is the first option, direct memory based question it is. Next question, choose the correct set of statements regarding population interaction between two species. This is a question from ecology. So here they are asking correct statements. In both parasitism and commensalism, only one species benefit and other species is harm. In parasitism, yes, it is correct. One species is benefited, other is harm. But in commensalism, one species is benefited, other is neither benefited nor harmed. So this option is wrong. Next, both species benefited mutualism. Yes, mutualism, there are so many examples where we have both the species are benefited. Next, both the species benefited commensalism. Again, the same one they are giving where one species is benefited and another species is neither benefited nor harmed. In emensalism, In parasitism, only one species benefit and other species is harm. This also is a correct one. And in amensalism, one species is harmed and other one is unaffected. In amensalism, one is hurt, another one is unaffected. This is also correct. So they are asking which statements are correct. They are asking. So first one is wrong as well as third one is wrong. Rest all are correct. That is B, D and E. So we can go with the fourth option. So this completes the zoology part. Next, Vamshi sir will start with the chemistry part.
Thank you, madam. Now let's see the chemistry part, how the questions are. As it takes a few seconds, I will give you the overall idea of the chemistry paper, how the questions came. Right. Majority of the questions are very well balanced between physical, organic and inorganic. And organic and inorganic can be done very much comfortably through NCRT support. So that is what the reason from day one onwards we were telling, and we made the students to revise it again and again. Almost 8 to 10 times the students have revised the NCRT textbook, and physical as usual some numerical questions are there that little time will be taken. So let's quickly get into the question discussion here. Coming to the first question, coming to the first question, is an question related to alcohols, phenols and ethers chapter. According to NCRT pattern we have classified. So in this question you have cumin, right? So cumin is converted into phenol. So that is what a named reaction, cumin hydroperoxide reaction. Cumin hydroperoxide reaction. So for cumin hydroperoxide reaction, what is the reagent you have to take? You should treat cumin with oxygen, O2 molecule to get converted into cumin hydroperoxide, which on hydrolysis will form phenol plus acetone. Right, phenol plus acetone. So therefore, option A is second one. Then coming to the second B molecule, B reaction, acetic acid to be converted into ethanol, right? Acetic acid to ethanol means a reduction reaction. So where is the reduction reaction? Here it is, this is there. So but what he has done is, first he has taken acetic acid, converted into by reaction with the methanol in acidic medium. Esterification is done, right? So converted it into CH3 C double bond O CH3, and this ester is reduced by catalytic hydrogenation to ethanol and methanol, right? Ethanol plus methanol. So this is a second option, that is 3. B is 3. So A is 2. So first option wrong. A is 2. B should be 3. Second option also wrong. A is 2. B is 3. So this is correct. And even in fourth also it is there. So let us decide between these two. Let's come to C. In C, what he has done? 1 propanol is converted into 2 propanol. How that is possible is 1 propanol first to be converted into propene, then to be converted into 2-propanol. So that is possible by first dehydration and then addition of water. So where is the dehydration? First concentrated sulfuric acid plus a heating, and then hydrolysis is done. So for C it is 4. So this is not the answer, this is the answer. So fourth answer is correct. Easy only, right? But it takes time. Then second question, the measure product Z formed in the following sequence of reactions is, first C2H6 is there, that on monochlorination you will be getting C2H5Cl, which on reaction with ammonia you will be getting C2H5NH2, which on diazotization followed by protonation, right? So you will be getting C2H5OH, alcohol, right now? So second option is correct. So this question was from hydrocarbons and amines mixed reaction. Then coming to the next question, number of chlorine atoms present in the organic compounds X and Y of the following reactions respectively is. What is this reaction? Benzene is treated with chlorine in presence of Lewis acid AlCl3. So it is a aromatic electrophilic substitution reaction. So first what will happen to the benzene? One chlorine will come, and as Cl2 you are taking 6 moles. What will happen? First with 1 mole you are getting chlorobenzene. With the second mole you will be getting, as chlorine is a ortho para director, you may get the chlorine here or here, ortho position or para position. Then again that dichlorobenzene will undergo again chlorination to trichlorobenzene, tetrachlorobenzene, pentachlorobenzene and hexachlorobenzene, that ultimately this will get converted into this compound. So hexachlorobenzene is what we will be getting. Coming to this compound, benzene with Cl2, 3 moles in presence of sunlight. This is free radical addition reaction, FAR, free radical addition reaction. One Cl2 molecule will be added here, second Cl2 molecule here, third Cl2 molecule here, that you will be getting the product here is aromatic character will be gone because it is a addition reaction, but same 6 chlorine atoms will come here also, right now. So in this compound 6 chlorine atoms will come, and in this compound also 6 chlorine atoms will come. So second option is correct, right? Then coming to the next question, in the following reaction sequence, X and Z respectively are. X and Z respectively are. First of all, what is this one? There is a 1 propanol which is treated with PCl5, right? So you are getting 1 chloropropane plus X. X is the byproduct here, phosphorus oxytrichloride, POCl3. So X should be POCl3. So this is not the answer. This is not the answer. Between 1 and 4, your answer should be. And coming to this propane, 1 chloropropane on alcoholic KOH. This is E2 elimination reaction. So you should get propene, right? So where is propene present? This is the propene, CH3, CH double bond, CH2. This with HBr in presence of peroxide. That is anti-Markovnikov addition, Karash effect, that the Z molecule you will be getting is this one, right? So CH2Br, 1 bromo propane. So this is what the 1 bromopropane. So here 2 bromopropane is given. This is a Markovnikov addition product. So 1 bromopropane is correct. Therefore, fourth option is perfect. Right. So going to the next question, identify the correct statement about ClF3 from the following option. First of all, let us work out on this is actually chemical bonding related. He is asking about the geometry, number of lone pairs and all. So first let us take out the hybridization of this central atom chlorine. Chlorine is the atom halogen having 7 valence electrons, 4, 5, 6, 7. So from the 7 valence electrons, 3 fluorines will form 3 single bonds, right? So the remaining 4 electrons present in the form of 2 lone pairs, right? So steric number here is 3 sigma bonds plus 2 lone pairs. Steric number is 5. So if steric number is 5, what is the hybridization? sp3d. sp3d hybridization. And in sp3d hybridization, chlorine 1, 2, 3, 4, 5. These are the 5, right now. So in this one, there are 2 lone pairs. Always the lone pairs will be occupying the equatorial position. So here 1 lone pair will come, then here 1 lone pair will come, then here 1 fluorine atom, here 1 fluorine atom, and here 1 fluorine atom. So this is T-shaped molecule, right now. So it has T-shaped structure, T-shaped geometry with 2 lone pairs with 3 lone pairs. 2 lone pairs is only correct. Therefore, option 2 is the perfect answer, right? So going to the next question. Select the reagents that reduce nitrile. What is a nitrile? Nitrile is RCN. Cyanide IUPAC name itself is nitrile. This nitrile to be reduced into RCH2NH2. RCH2NH2. Right. So in this one with lithium aluminum hydride, I can convert cyanide to nitrile, cyanide to amine. Then tin plus HCl also will do the same function, cyanide to amine, Even H2 plus nickel also can be used in conversion, right now. So all three options are there in first one. This is first option is correct, right? So going to the next one, in option D, sodium amalgam plus ethanol, that is for partial reduction. Bromine plus aqueous NaOH sorry, bromine plus aqueous NaOH is not for the reduction reaction. Okay, na? Then going to, during a lessons test, the elements present in an organic compound, are converted into inorganic compounds, ionic compounds. Right? Suppose let us assume there is a compound like C2H5CN is there. Cyanide only, just before we have seen. Na? So this compound, when it is treated with sodium metal during lessons test, it will be converted into whatever the nitrogen atom is present, which is in covalent bonding. Covalent bonding will be converted into ionic compound Na plus Cn minus, ionic compound. So covalent form to be converted into ionic form. Covalent form to be converted into ionic form. Understanding? Na? So very simple example. Suppose you take CH3Cl is there. When it is treated with sodium, this Cl whatever is there will be converted into NaCl, like Na2S, sodium sulfide. Like that, all the organic form, covalent form elements are converted into ionic form during lessons test. Coming to the next question, this is also from the some of the basic concepts and techniques in organic chemistry with respect to NCERT. I am talking about, right, the pair of molecules that are metamers among the following is, one is this is alcohol, this is also alcohol, but OH group is in the first position, here OH group is in the second position, so they are positional isomers. And here this is CH3O C3H7, and coming to here C2H5O C2H5, these are the metamers, right? And here it is a ketone, here it is aldehyde, therefore functional isomers. And coming to this one, all the 5 carbon atoms are present in the straight chain here, and here one of the carbon atom went into the branching, so these are chain isomers. Therefore you can understand only second option is the one which is the metamers, right? So going to the next question, this is also related to chemical bonding only, just he is asking how many sigma bonds, pi bonds, lone pairs are present. First if you see C2H4, right, C2H4, C double bond C, H, H, H and H. How many sigma bonds are there here? 1, 2, 3, 4, and fifth sigma bond is there between 2 carbon atoms. Total 5 sigma bonds are there, right? So C2H4 with 5 sigma bonds and 1 pi bond. So option A to have 4, right? Option A to have 4. So remaining all can be eliminated. You got the answer directly. Remaining also you can solve it in the similar manner, right? Going to the next question, compound P C8H8O gives a orange red precipitate with 2,4-DNP. What is 2,4-DNP? That is the reagent used for identifying carbonyl functional group. Maybe a ketone or aldehyde also, but carbonyl functional group, right? So carbonyl functional group is there. Next, it does not reduce a Fehling's reagent. Not reducing Fehling's reagent, you can understand that it is not a ketone, right? It is, sorry, if it is reducing Fehling's reagent, it will be an aldehyde. It is not reducing Fehling's reagent, it should be a ketone. Even aromatic aldehydes also will not reduce, right? So it should be a ketone. Let me assume like this, right now. And then on drastic oxidation with chromic acid, that is a H2CrO4, P gives an aromatic product Q that produces effervescence on treatment with aqueous NaHCO3. In sodium bicarbonate test, this compound Q is liberating carbon dioxide. Who is that compound Q? Nothing but benzoic acid. Benzoic acid. Q is benzoic acid, right? That produces effervescence with NaHCO3. Compounds P and Q are So with this, what we can understand is whatever the C8 is there, already one carbon is over, remaining 7 carbons are there. This side you have to take a 6 and this side you have to take 1, right? Na? So the compound will become benzene with a C double bond O and left over one methyl group. So it is acetophenone. So this is acetophenone, remove this one, option 3 gone, option 4 also gone. Now coming to Q, this is not the compound, this is Q, what we got. So this is correct. So remaining all are wrong. So second option is correct. Easy question only, little lengthier, but still definitely we can do it. Then functional group that can be identified through phthalate dye test. It is what phenolphthalein indicator will be treated, will be prepared here, right now. So phthalic anhydride with 2 moles of phenol in presence of sulfuric acid on heating will undergo dehydration to form phenolphthalein indicator, right? So this is what is the, here typing mistake, it is phenol. Right. Then going to the next question, the correct statement with regard to secondary structure of DNA and RNA. Biomolecule chapter, it is very simple question. It should be DNA possesses a single stranded helix and contains uracil. This is wrong because uracil is not the nitrogenous base present in DNA. It will be present in RNA. Then RNA possesses a single stranded helix, up to here it is okay, and contains thymine. This is also wrong. RNA will have uracil in place of thymine, right? DNA will have adenine, guanine, cytosine, thymine, whereas RNA will have adenine, guanine, cytosine and uracil. In presence of, in place of thymine only, uracil will be present. So this is also wrong. Now coming to DNA possesses a double stranded helix, this is correct, and contains thymine, this is also correct. So option 3 is the correct answer. Going to the next question, the IUPAC name, correct IUPAC name, very simple it is. First let us understand the longest carbon chain to be the parent chain, 1, 2, 3, 4, 5, 6, 7. Even from here, you go here also 6, 7. So it is same, either you come straight or go up, right? So if I am taking straight, 1, 2, 3. At third position, methyl is there, and from this side, if I number, at third position again, ethyl is there. So from which side I have to give numbering, left to right or right to left, depends upon alphabetical order. Here it is starting with methyl M, and this is starting with ethyl E. So E comes first in alphabetical order. Therefore, numbering from this direction will become correct, right? So it will be 3 ethyl, 5 methyl heptane. So very simple question, very direct question, right? So going to the next question, see, it is from the coordination compounds, easy question only, nothing to calculate, nothing to see here, just ligands you have to observe in this question, right? So first one, what he is asking, PtCl2NH3 taken twice is given. This compound, is it octahedral, trigonal bipyramidal, square planar, tetrahedral? That means the geometry you have to mix with it, right? So here all platinum compounds with 4 ligands, square planar, right? So A should be with 3. So A should be with 3. This is not these two. Between these two, answer should be. And coming to CONH3 taken 6 means 6 ligands are there. So it should be octahedral. So B should be 1. So which is the B1? Option 4. Simple, right? Going to the next question. The following 2 reactions give the same foul smelling product. What is the compound which gives the foul smell? That is isocyanide, right? RNC. So here first it is C2H5Cl. In presence of X, it is giving Z, right? Z is what? The isocyanide. So which compound, which reagent on reaction with C2H5Cl will form isocyanide? That is, when you are taking RCl on reaction with KCl, right? 2 different reagents are given here, KCl and AgCl. KCl is an ionic compound, K plus C triple bond N, right? But it is a species in which 2 donor atoms are present. On carbon, lone pair is there. On nitrogen is also lone pair is there. So carbon is the one which will donate electron pair because carbon less electronegative, holding the negative charge unfavorable, therefore it will try to donate it faster. So this becomes the donor atom. So C triple bond N, that this will be donated and Cl minus will come out, that you will be getting RCN. Okay na? So KCN is not the X. KCN is not the X. Now you have to opt between first and fourth options, that is AgCN. AgCN is there, and who will give the foul smell? Cyanides will not give the foul smell. It is the compound isocyanate which will give the foul smell. Therefore, option 4 is correct. Right. So this is what the first 15 questions I have taken. Now I welcome our another senior most chemistry faculty, Nikhil sir, to continue with the remaining questions. Sir, please.
Thank you, sir. So let's start with the next set of questions. So let's start with the first question now. What is it? Qualitative analysis Bi plus 3 is detected by the appearance of a solid. Calculate the pH. Now this is a question of ionic. And in this, we will be mostly discussing physical and a few bit of organic also. So this will be a little bit lengthy for calculation part. What we have, they have given us the equation for this. At high equilibrium, what they are asking us, calculate the pH. So what we will write? We can write concentration of, if I write K will be what? BiO plus into OH minus, and as they are both equal in number, so let us say this number is X, so this number will also be X. So what we will get from here? 4 into 10 to the power minus 10 equals to X square. So X value you will be getting as 2 into 10 to the power minus 5. So this, what is this X? This is nothing but OH minus. So as you have got the concentration of OH minus, can we directly calculate POH from here? What it will be? It will be minus log of OH minus, and that will be minus log of 2 into 10 to the power minus 5. So it was a little bit calculating question, but answer will be very simple from here. 5 minus log 2, as they have given the value of log 2 at 0.3 approx. So what we will be getting here? Approx 4.7. This is POH, but they are asking about pH. So pH will be what? As we know, pH will be 14 minus POH, that is 14 minus 4.7. So answer will be 9.3. So first question was from ionic and its answer will be option number 3. Let's move to the next question. Now, when 1 decimeter cube of CO2 gas is passed over hot coke. Now this is a question of idiometry of mole concept. A simple question. What they are saying, we are passing CO2 over carbon, right? And what it is giving us? It is giving us a gaseous mixture. After complete reaction, it becomes 1.4 decimeter cube. So can I say CO2 plus C? What it will give you? It will give you twice of CO. So if I have one molecule of this, or what we have here, 1 decimeter cube. So 1 decimeter of cube, how much it will break? Let us say X amount of this will break. So if X amount breaks, CO will be formed at 2x. So total volume, final volume will be what? 1 plus 2x minus x, and that is given as 1.4 decimeter cube. So on solving, 1 plus x equals to 1.4. So x will be getting as 0.4 decimeter cube. So at the end, what will happen? We will have 1 mole, we had 1 mole of CO2. How much it has dissociated? 0.4 has dissociated. So how much it is left? 1 minus 0.6, 4. That means 0.6. So CO2 will be 0.6 and CO will be 0.8. So now let's check which option it is. Option number 3, 0.8 decimeter cube of CO and 0.6 decimeter cube of CO2. So this will be our second option. Third option will be the correct one. Now third question, a very easy question from atomic structure. We have directly to find orbital 2 1. What we write for S, L value is 0. For P it will be 1. For D it will be 2. And for F it will be 3. So first one is n given is 2. What is n representing? Principal quantum number. So 2 1, that means 2 with P. So what it will go with? Second one. Then we have 4 0, that means 4s. So A will be 2 and B will be 3 and 5 3, that means it will go for 4 and D will be going for 3 2, that is 1. So which option is correct here? 1 A2, B3, C4 and D1. First option will be the correct. A very simple question, good scoring question. Now let's move to the next question, that is of electrochemistry. What it says, we have to calculate the EMF of, they have given as hydrogen electrode, right? So we can just apply Nernst equation. Simple application of Nernst equation. What it was? E equals to E naught minus 0.059, what they have given here, by n log of Q. Now what is interesting, they have asked us EMF, that means they are asking for SRP value. So if they are asking for SRP value, what equation I will write? H2 H plus will break to give you H2, right? And how many electron will be gained here? 2 electrons. So n value will become 2, and what I will write for Q? Q will be pressure of hydrogen by concentration of H plus square. So we can put it over here. What is the E naught value for a standard hydrogen electrode? It is 0. So it will be 0 minus 0.059 by 2 log of pressure of hydrogen. What is given as pressure of hydrogen is 2 atm and it is given as 0.02. Okay na? So from here we can solve it, and what we will be getting at 0 minus 0.059. Now it will be coming at 4 into 10 to the power minus 4. 2 we will divide it, log of so this whole value, if we are going to solve, we are getting as minus 0.109 volt will be the value. So option 4 will be the correct one. Next we move to the next question, that is now a simple question on kinetics, unit of rate constant. What it is? R equals to rate of any reaction will be equals to K concentration of the reactant to the power of order. So K, what we can write? K will be equals to rate, that is mole per liter per second divided by concentration, that is mole per liter to the power of X, where X is the order of the reaction. So as we know 0 order, if you put 0 over here, what we will be getting? Mole per liter per second. So for first we will be getting as 4. For first order we will be getting as 3. If you put 1 here, we will get per second as the normal answer. So for A it is 4, for B it is 3. I think we can solve from here only. A4, B3 no. Second order, let's put order value 2. What I will be getting here? Mole inverse liter second inverse. What it is? 1. So mole inverse liter per second. And third order reaction, if we put 3 over here, what I will get? Mole minus 2, that is second. So D will be 2, D2. Option 3 is the correct one. A4, B3, C1, D2. Let's move to the next question. Atomic structure portion, and now it will be a little bit lengthy for the calculation. Why? They have given as a 150 watt ball. What is watt? That is joule per second. That means power that is given for the bulb, that is 150 watt. It is producing 150 joule per second, and it is converting 8% of energy. So energy that is emitted will be 150 into 8 by 100, that is 12 joule. Okay. Now, if energy of one photon is this much, 4.42, 10 to the power minus of 19, how many photons are emitted? So, what we can say, if n photons are emitted, for one photon energy they have given as 4.42, 10 to the power minus 19. So, for n moles, how much it will be? 4.42, 10 to the power minus 19 into n. And this value will be equal to what? This value will be equal to energy emitted. So what I can say, N will be equal to 12 by, now we can solve it completely, but that will take some time. What we can do, we can cross check with the options. It will be giving us 10 to the power 19, right? And 12 divided by 4.42, it will be something approx 2 point something. Why? Because if it is 4, it will become 3. If it is 5, it will become 4 point something. So it will be more than 4.2.5. So if we look at the option, you can just cross the option and we can get the answer. Why? Because we have less time, and for solving for the complete calculation, it may be time taking. So I will go by crossing the options. Next question. They have given number of hydrogen atom present in 5.4 gram of urea. Mole concept question, very easy question. Urea, what it is? NH2, CO, NH2. Okay, and molar mass they have given as 60. The number of moles of urea, what you will be getting? Given mass by total mass. That means, sorry, molar mass. So what I will get? 0.54, sorry, it is 5.4. 5.4 divided by 60. So number of moles of urea will be how much? It will be 0.09. And each mole of urea has 4 hydrogen. So number of hydrogen atom, if they are asking, hydrogen atoms they are asking. So number of hydrogen atom will be how much? 0.09 into 4, that means 0.36 mole. So we can multiply it by 0.36 into 6.023 into 10 to the power 23. Now again we can solve it completely, otherwise we can cross check from the options, and 3.6 into this will be approx 2.16. So I am not going to completely solve it. From the options I am getting this and this, but here it is 10 to the power 22. And what we are getting? 10 to the power 23. So option 1 will be the correct option. So in this question and the last question, what we have seen, we can go for complete solving, but to save time, you can just cross check from the options that is given, and that can save your time. Next question is from thermodynamics. What it says, a simple question on the first law of thermodynamics. What is first law of thermodynamics? Delta U equals to W plus Q. And what they have given, 500 joule is observed, and system. Now this is the interesting thing. 200 joule is work done is by the system. Work done by the system. That means that is a expansion work. And being expansion work, it will be negative, right? So if I put it over here, so del U will be how much? 500 is total energy that is absorbed, that will be positive, and energy of work done, that is negative work done, will be how much? 200, but it will be minus 200. Why? Because it is expansion work. So what you will be getting as 300 joule. So option 2 is the correct one. Simple question from thermodynamics, and it must take very less time. Next question is also very simple, direct question. From a hydrocarbon, methane reacts with steam at 1273 Kelvin. What it gives us? This will be give us water gas. What is that? CO plus H2. So first product that we get from methane plus steam, that is CO plus H2. So option 1 will be the correct one. A solution of copper sulfate is electrolyzed for 10 minutes with a current of 1.5 ampere. The mass of copper deposited at cathode. Again, a very lengthy calculation from electrochemistry, from first law of Faraday. What does it say? Weight deposited will be equal to ZIT, that is E by 96500 into I into T. What is E? E is equivalent mass. I is current and T is time in second. So we have copper sulfate solution. The solution that you have taken is sorry, CuSO4. Charge over here is +2. So N, number of electrons gained or lost will be 2. So equivalent weight will be molar mass, that is 63, that is given. Actually it is 63.5, but we will go by the value that is given in the question. N is 2. So E will be 63 by 2. Now if I put here, W will be equal to 63 divided by 2 into 96500. Now current what they have given is 1.5 ampere and time they have given is 10 minutes. So now I have to convert into second. So it will be 60 into 10. So we can solve it from here. Now again we have to completely solve it, or we can just check what can be the approximate answer. If we solve it completely, why? Because many things will be cut. What I am getting? 63 into 3 into 1.5 by 965. So this is the value that we will be getting, and we can also cut it from here. 193 0.3. So it will be 63 into 3 189, and approx it will be 0.3, approx 0.3. Again I am going for the approximation. Why? Because the options given are having large gap between them. We can go for complete calculation, but 63 into 3, that is 189. Approximately they will be cutting out each other. You will be getting something below 0.3. Which option that is? That is the fourth option. So now by this method that we are doing in one or two questions, we can save some time here. Now let's go to the next question. Identify the correct statement. The first question is molality of 2.5 gram of ethanoic acid in, there is some printing mistake over here, it will be 75 gram of benzene is this much. What is molality? Molality is nothing but number of moles. Ethanoic acid, what it is? CH3COOH. So what is the molar mass here? Molar mass will be I think 24 and 32 56 and 4 60. So molar mass here is 60 gram. So number of moles will be how much? 2.5 by 60, that is number of moles, divided by weight of solvent in kg. So divided by 75 by 1000. So I am writing it as 1000 by 75. What will we are getting as 30. So you are getting as 10 by 18. 10 by 18 is 0.556. Yes, this is a correct statement. The molarity of a solution containing 5 gram of NaOH. What is the molar mass of NaOH? 40 over here. In 450 ml of solution is this much. Directly we can calculate molarity will be number of moles, that is 1 by 8 divided by volume of solution in liter. So again we have to calculate it, and what we will be getting? So we are getting as 20 by 72. That will be approximately how much? Approx 2.8. 2.8. Yes, it is correct. So option B will be also the correct one. Third one, aquatic species are more comfortable in cold water. Now it is the application of Henry's law. What happens when the temperature increases, then the oxygen that is dissolved in water will decrease, and hence that is called as thermal pollution, that will make aquatic species uncomfortable. So aquatic species are more comfortable in cold water. Why? Due to more oxygen dissolved in them. So option C will be the correct one. Now option D, solubility of gas increases with decrease in pressure. No, just opposite of what Henry law say. So it is just a theoretical thing. Wrong. For a binary mixture of A and B, number of moles of A and B are this and this. The mole of fraction of B will be what? NB by Na plus Nb. Given mole by total number of mole. This one is also correct. Incorrect. So A, B, C. Option 2 will be our correct answer. Now let's move to the next one. Solution and colligative property. Normal question they have asked and direct question they have asked. Chloroform and acetone. What is chloroform? CHCl3. And what is acetone? CH3, C double bond O, CH3. Now it is an interesting question. Why? Because we have some application of bonding also over here. After mixing, what happens? CH3, C double bond O, CH3 and CHCl3. Now here, due to the very high minus I effect of CCl3, H will have a very high positive charge, and with the lone pair over here, they will have intermolecular H bonding. Now it is also an interesting fact. Why? Because generally what we say, कि for H bonding we need fluorine, oxygen and nitrogen, but 3 chlorine on a carbon will generate similar positive charge, and hence there will be intermolecular H bonding, due to which it will show negative deviation. So which option will be correct? Formation of H bonding between acetone and chloroform. So this one will be the correct answer. Let's move to the next question. Buffer solution. Now again one question from ionic. What it says? We have equal concentration of X and HX. It should be X minus, a small mistake over here. So X minus and HX. What we have in it is an acidic buffer. And if in an acidic buffer, if we have same HX and the salt, concentration of X minus and HX is same. So what it will be? It will be the best buffer. For best buffer, pH value is what? pH value equals to pKa. Right? What they have given us? They have given us Kb for X minus. Now, what we have to do? We have to find out Ka from given Ka. What we know here? Ka into Kb will be 10 to the power minus 14. So, Ka will be 10 to the power minus 14 by Kb. What is Kb over here? They have given 10 to the power minus 10. So, what we will be getting as Ka 10 to the power minus 4. So pKa value will be 4, and that will be same as that of pH of this solution. So option 3 will be the correct one. Next question. Let's move to the next one. Question from kinetics, direct question that we study in the graph, and that is we have concentration of reactant versus time. It is a constant graph. For which order it is? We can say directly from 0 order. Why? Because we write AT equals to A naught minus of KT, that is Y equals to minus MX plus C, where A naught is our C and slope will be equals to minus of K. So it is a direct question and a very easy question from kinetics. So it will be 0 order reaction. Option 1 will be the correct answer. Now let's move to the next question. Consider the following reaction, 2A plus B forming 2D. They have given us delta U and delta S. Identify the correct option of delta G. Now, a lengthy question, a calculative question from thermodynamics. What we have to do? We have to find delta G. What is delta G? Delta G of any reaction is delta H minus T delta S. They have given us delta S, but delta H they have not given, and for that we have to calculate from where? Delta U. What we know? Delta H minus delta U equals to del Ng of RT. So delta H we can calculate from here. What I will get? Delta H will be equals to delta U. How much it is? Minus 10 kilojoule plus del Ng. For this reaction, we have 2 moles, 1 mole and 2 mole. Del Ng will be how much? 2 minus 3. So it will be minus 1 into R into T. If you look, every value they have given in SI. So we are using it 8.314 and T will be using as 298. So again, if we need, we can solve it completely, or we can just take it to be 300 approximate, just to find out whether it will give us a positive or negative answer. So it will be something around 1224, so it will be minus 12 point something value you will be getting from here. Now we can apply it from here. Why I am doing this? Because we have to just check whether it is positive, is non-spontaneous, or negative, is spontaneous. So delta G will be what? That will be delta H minus of T delta S T delta S. Okay, just 1 minute. Yeah. Okay, sorry, let's continue with this. T delta S. Now let's put the value. T delta S will be how much? They have given us delta S as 44 and temperature as 298. We can directly apply 44 into 298. Now if you look at this value, we can calculate it completely, but it will be something around 13, right? 44 into 300. 13 point something. That means whatever value you are getting from delta G, that will be positive and that will be less than 1. Why? Because it's 12.4 approx and it is 13.1 or 2 approx. So what option we have is it can't be there. It is non-negative. It is positive but less than 1. So option 2 we will go with. This will be the value for delta G and it will be non-spontaneous. Now let's move to the next question. Given is a rate constant, again a lengthy question from kinetics. And what we can say, what we have, k equals to a e to the power minus of ea by rt. So ln of k will be equals to ln of a minus of ea by rt. So if you compare this ln k, we have got the same value. So what we are getting, ea by r value will be how much? Minus 1.25. Minus will cancel out. So what we will be getting, EA by R will be equals to 1.25 10 to the power 4. Right? Again we have to solve the complete one. 1.25 10 to the power minus 4 into R. What is the value of R? Approx 1.978 or 98 they have given in. Now again, if we need to calculate this whole thing, it will take some time. If you want to save time, what we can do? We can take it approximately 2. What we are getting over here? We will be getting something as 5,000, right? Sorry, 2 into 25,000. Approx, if I am taking 2, so answer will be something close to 25 kilojoule. Let's see the option. Do we have it? No, no, no. What is the best option? So 4 or 5 questions of this topics of physical were lengthy, but if we need to solve them, we can approximate it by the options also. So that's all for the physical part. Now another ma'am, Komal ma'am will be going on with the next part, next 14 questions that is left. So ma'am.
Thank you so much, Nikhil sir. That was a great insight of all the physical chemistry questions, especially with the calculation part, right everyone? Okay. So this is your Komal ma'am, and first of all, I congratulate all the NEET students for writing a really big and noble exam, right? And this exam is going to give you a really insight that how to do a hard work with a smart work. Correct, everyone? Okay. So now I'm coming with all the inorganic questions. This time we have seen the relative. I have seen what similar number of questions for inorganic, physical and organic. So all were having the more or less same weightage only, organic, inorganic and physical, right everyone? Okay, so let's start with the first question. I'll be very quick with the physical chemistry. Sorry, inorganic chemistry, I'll be really quick with that. So coming to the first question, list 1 and list 2 match the following question. Very important, and such type of questions are coming really like too much in the exams nowadays. So B2O5, what it is saying? So they have given the compounds, complex or transition metal one side, and the next side they have asked the catalytic role of that. So V2O5, very simple. As soon as I listen to V2O5, I know it is going to convert SO2 into SO3, correct? And that is further going to use to convert it into the H2SO4. Yes or no? Right? So V2O5 is preparation of H2SO4 from SO3. So I must say option A is option 3. Option A is matching with option 3. So either my first or fourth option is correct. So I got the eliminated. I eliminated 2 and 3. Fe. Now coming to the Fe, let's see preparation of ammonia from N2H2 mixture. Yes, Haber's process. N2 plus H2 is giving us NH3. Haber's process use Fe. That means option B is matching option 1. Option B is matching option 1. I got my option only with the two answers. Only with the first two options I got my answer. But still, let's see. PDCl2. PDCl2 oxidation of ethyne to ethanol, correct. Ni complex polymerization of alkynes, correct. Right, everyone? Okay, so this was a very easy question coming from D and F block element. Coming to the next question, very easy question. Spin only magnetic moment Ti plus 2, and see, you don't have to worry about even the configuration. They have given you the configuration as well. Still, I want to tell you the configuration. Titanium is atomic number 22, right everyone? So it will be what? Argon, 4s2, 3d2. Now when I go with Ti plus 2, when I go with Ti plus 2, it will be argon as it is 4s0, 3d2. So 3d2, how many unpaired electron will be there? 2 unpaired electron. 2 unpaired electrons, number of unpaired electron will be 2. How to find magnetic moment? The formula is root 2 n plus 2. Now I know the n value is 2. But eventually I told you the trick also, that is whatever be the value of n. Suppose n is 2, so just add to 7.7. 5. Just add 0.75 and approximately you will get the magnetic moment. So which value is matching the nearby value of this one option? So I guess option 1 is matching the nearest to my option. So 2.84 Bohr magnet is the correct answer. I guess every one of you got it right. Two products X and Y are formed in the following reaction sequence. Table for the preparation. So organic chemistry plus separation method is mixed in this question. Okay, now organic chemistry, which part is mixed in this question? Benzene with CH3Cl chloroform. Benzene with chloroform, what is going to do? Electrophilic aromatic substitution, where hydrogen will be substituted with the Cl, right? So it is, CH3 positive, where H is going to be replaced with the CH3 positive. So what is your W? W is your toluene. What is your W? W is your toluene. Now this toluene, when further reacted with dilute HNO3, what is going to happen? Nitration in the presence of H2SO4. Nitration will happen. Now it is saying something, you are getting X and Y. So what? CH3 is electron donating group. Electron donating group is activating what? Your ortho or parasite. So NO2 will come at ortho position, as well as NO2 will come at para position, right? So these are the product X and Y you will be expecting here. These are the X and Y product you will be expecting here. So this is your WXY. This is your WXY. Now it is asking the suitable method of separation. Suppose in a mixture I am having WXY all three. Separation method, very easy, fractional distillation, because of the difference in boiling point. The difference of toluene, the boiling point of toluene is really less as comparative to the X and Y. So fractional distillation easily can be done to separate them out. So option 3 was the correct answer. Right? In a test tube containing a salt, a few drops of dilute H2SO4 was added. Give colorless vapors having smell of vinegar. Now the question ends here. What is it saying? Smell of vinegar. Let's see, but the vapor turns the blue litmus paper red. Blue litmus paper red means it is going to be acidic in nature. Identify the correct anion from the following. Now it is showing me it is a smell of vinegar. Smell of vinegar means acetic acid. The smell of vinegar means acetic acid. Right? So salt, when it's going to combine with H2SO4, it is going to give me acetic acid. So acetic acid must be coming from the acetate ion, CH2CO negative, because how do I know that acetic acid is formed? First of all, vinegar smell. Second, it is acidic in nature, which is also seen by the litmus paper. So I am knowing that what carboxylic acid is forming, that is CH3COH, acetic acid I am getting. And for acetic acid, which anion I needed? Acetate ion I needed. Right everyone? Very easy question. As soon as I learned the vinegar, I got the answer. Identify the incorrect statement of the following. So this question is coming from the periodic, or you can say chemical bonding mix. Carbon has the ability to form p-pi multiple bond. Yes, you have seen carbon carbon, carbon triple bond carbon, right? So carbon can make p-pi bonding. Correct statement, but we have to identify incorrect statement. This is the kick here, right? PCl3, Cl3, not H3. Chlorine is there, not hydrogen is there, right? So boron and aluminium. It is saying if E is boron, then it is monomer, and if E is aluminium, then it is dimer. Correct. Very, very, very correct. Right, everyone? Okay. So oxygen exhibit only -2 oxidation state. You have seen it and you have taken it. So no oxygen has -2, sometimes -1, you call it peroxide, and so on. The order of catenation property group 14, obviously catenation is famous for group 14, especially carbon. Okay. So going to the next question, although +3 oxidation state is most common in lanthanoids, cerium is special +4 oxidation state. So what is the configuration of cerium? right? The cerium is somewhat like Xc, 4f1, 5d1, 6s2. Can you see how many electrons in the outermost shell? 1, 2, 3. So when it is going to be a +2, it is getting a nearest noble gas configuration. It is getting a nearest noble gas configuration. So nearest inert gas is xenon? No, xenon. After losing one more electron, it acquires +14? No. F4f0, it is getting, the atomic number will be 61? No. After losing one more electron, it acquires 4f0 electronic configuration? Correct. It will acquire the 4f0 electronic configuration. So option 4 is the correct answer. I told you through the configuration how. Identify the incorrect statement from the following. Now it is saying phosphorus compound and arsenic compound form d pi bond with transition metal. Yes. Why? Because phosphorus arsenic having the availability of d orbital, so they can form d pi d pi. Nitrogen can form d pi bond with oxygen. This is absolutely wrong. Why? Because nitrogen is only up to 2p. It does not contain d orbital. Because it does not contain d orbital, how it is going to make a bond, right? And the question is asking the incorrect statement. So this looks to be incorrect, but let's see the other options as well. Nitrogen can form p pi multiple bond with itself. Yes, N2. Phosphorus arsenic entities show catenation property. Yes, that's why you see P4, right? Okay. So option 2 is the correct answer in this case. Option 2 is the correct answer. Now, qualitative analysis, we are seeing the question. Phenolphthalein indicator is used as an indicator for a titration of sodium hydroxide solution. Again, a standard solution. Standard solution, where standard solution is put in burette, right? And the other solution will be in the conical flask. The color changes that is observed at alkaline pH. So phenolphthalein in acidic medium is what? Colorless, and in basic medium it is pink. So we are going from acidic medium to basic. pH alkaline, we are going towards the alkaline pH, right? At equivalence point. So that means we are going from acidic to basic. Acidic means colorless, basic means pink. So colorless to pink. Option 3 is the correct answer. Option 3 is the correct answer, right everyone? Okay, going to the next question. Identify the incorrect statement from the following. The high-effect name of the element with atomic number 107. What is 1? Un. What is 0? Nil. What is 7? Sep, and we ended with em. So yes, the first is correct. Again, the question is asking the incorrect one. Are you seeing the trend? They're asking the most of the time they're asking you the incorrect one. So the largest and the smallest species among Mg and this one. So largest species among Mg is Mg, and the smallest species in both of this will be Al3 positive. But they have given us the wrong answers, right? So option 2 comes out to be incorrect. But let's see 3 and 4. The similarity in behavior of lithium and magnesium is because of diagonal relationship, correct? The oxidation state and covalency of aluminium here is +3 and respectively. So yes, this is -1 and this is 2. When you calculate it, it comes out to be +3 for aluminium, and 6 and 1 is, sorry, 5 H2O and 1 Cl, 6, total 6. So covalency is 6, oxidation state is 3. So it is also correct. So incorrect option is option 2. Incorrect is option 2. Clear everyone? Going to the next question. The correct order of increasing metallic character. Now when I talk about metallic character, so here the elements are given to me is of the second period and of the third period. Which is given for the second period? Second period element I can see only beryllium. Third period is sodium, and I can see magnesium. Then group 14 is from silicon, and group 15 phosphorus I am having. Correct everyone? So this is group 1. This is your which one? Group 2. Now here it is 14. I am not writing the actual Roman numbers. Again I think I am not writing the actual Roman numbers. And here I am having what? Your group 15 here I am having. Right? Group 15 here I am having. So you should know top to bottom metallic character increases and left to right metallic character decreases. So who's having the least metallic character? I can say phosphorus is having the least metallic character. So phosphorus, so answer can be 1, 3, 4. Option can be 1, 3, 4. And here I can see most metallic character will be sodium. So sodium is having the highest metallic character. So 1, 3, 4. 1, 3, 4. Answer can be either 1 or either 4. So after phosphorus, I'll be seeing the silicon. After phosphorus, I'll be seeing the silicon. Right here it is magnesium. So I got my answer option 1. I got my answer option 1. So phosphorus, silicon, then magnesium, and then beryllium. Phosphorus, silicon. Sorry. We have to told the lower to upper. So phosphorus, silicon, beryllium, and then magnesium. Why first beryllium? Because I can see top to bottom it increases. So it is having the lower metallic character, it is having the higher metallic character, and then sodium. So option 1 is the correct answer. Option 1 is the correct answer, right? Which of the following is an impedanted ligand? Very easy question. First is ethylene diamine tetraacetate, EDTA. EDTA is your monodentate, that is your, gives you 4. Tetradentate. Oxalate, if I ask you CH3CO, that is your bidentate ligand, right? C2O5, C2O4, 2 negative. Oxalate is your bidentate ligand, right? If I talk about the ethane 1,2 diamine, again your bidentate ligand, but it's asking ambidentate ligand. Ambidentate ligand is thiocyanate, SCN negative, so it will be thiocyanate, and CS can also be there, right? So ambidentate ligand, if I talk about, so it is option 4. Option 4 is the correct answer. Option 4 is the correct answer. Going to the next question, the correct formal charge on the oxygen. So if I talk about the formal charge, valence electron, then minus non-bonding electrons, minus bonding electron divided by 2. So I am solving only the first one. If I talk about the second oxygen, valence electron 6, non-bonding are 4, minus bonding electron 1, 2, 3, 4. So it will be 4 by 2. So it comes out to be 6 minus 6, which is equal to 0. So this comes out to be 0. When you similarly find out, this comes out to be plus 1 and this comes out to be minus 1. So which is matching? It is asking the 2, 1, 3. So 0 plus 1 minus 1. 0 plus 1 minus 1. So you have to match this from here, right? So 0 plus 1 minus 1 is your correct answer. Option 2 is your correct answer. Going to the next one, given below the certain reaction, identify the reaction Kp equal to Kc. Very easy, right? Which formula you have to use? Kp equal to Kc R to RT to the power delta Ng. You only have to find the delta Ng value. Gaseous to both the sides, product minus reactant. So 2, 1 and 1. So 2 minus 2 comes out to be 0. So delta Ng comes out to be 0. Here Kp equal to Kc. So this is not my answer. I have to tell where Kp is not equal to Kc, right? So here I can see 2, 1 plus 1 is also 2. So 2 minus 2, again delta Ng will be equal to 0 here. If I look at the last option, 1 and 1 is 2. Here also stoichiometry 1 and 1 is 2. So 2 minus 2, again delta Ng is equal to 0. But if I look at here, 2, 3 and 1. So here it is 4, here it is 2. So 2 minus 4 comes out to be minus 2. So here for option C comes out to be KCRT to the power minus 2. So Kc is lesser than Kp. Kc is lesser than Kc. So it is not equal. Option 3 is your correct answer. Option 3 is your correct answer. Right, everyone? Okay. So coming to the last 45th question of today's exam. What is that? Coordination compound question. It is giving you very easy question again. PTNH3 whole twice Cl2. I think everyone of us knows this, that this is a square planar structure. Square planar structure only exists in cis and trans. Cis and trans means geometrical isomer. A is 3. A is 3, so answer can be either 1 or answer can be either 3. COEN post-rise. Bidentate ligand. Bidentate ligand, so it will be the non-superimposable mirrors. Non-superimposable mirror means it can be an optical isomerism. So B will be given in 4. Now coming to the CONH3 whole 5 and NO2. What is the ligand NO2 type? NO2 is a type of what? Ambidentate ligand. Ambidentate ligands will show you the linkage isomer. So C is 4. C is 4. I think I got my answer. But let's see the last one, H2 and Cl. H2 and Cl combinedly will give you what? Hydrated linkage, right? So that is what solvate. Solvate geometrical solvate isomerism you are having. So it will be D ka 2 answer. So option 3 is your correct answer. Right, everyone? I think this was the last 45th question of today's chemistry. So hope you all got it correct. So congratulations again to all, each and everyone. Now I'll giving to for the physics part.
Hello, good evening all. Right now, I guess you all are here. Check your physics key and solutions one after the other. Let us do each and every question analysis from the chapter, subtopic, what is the right key for the question, everything now here. Successfully, I guess all of them are excited. Check their key. Shall we start? Oh, now
Let's read the question. We know it's from moving charges and magnetism. You will have a detailed analysis of this once again once the paper discussion is done. So let's check what is given. He has given n number of turns. That is a circular coil which has a radius 5 centimeters and has magnetic field B. This is at the center. He is asking you to find out what is current and the magnitude of magnetic movement. These are the two questions which he asked us to find out. Let's say if you have a circular coil, what is the formula for magnetic field at the center of a circular coil? B is equals to mu naught I by 2R. But there are n number of terms. So you just have to put it before that. B value is how much? 3.14 into 10 power minus 3. N value is 100. Mu naught value we know 4 pi into 10 power minus 7 into current I we have to find out. By 2 into its radius is given as 5 centimeters. So you have to convert into meters. So 10 power minus 2. 3.14 is nothing but pi. We made the calculation so easy for us. 5 into 2 is 10. So this gives you 10 into 10 power minus 2 is 10 power minus 1. Send this to there. 10 power minus 4. This is again 2 zeros makes it 10 power minus 5. 10 power minus 5, 10 power minus 4. 10 divided by 4 is giving you the value of I. So what is the value of I? 2.5. How many options have I value as 2.5? Two options. Now how do you find magnetic moment M is equals to NIA. What is N? 100. I which is called 2.5 and area is pi r square. Pi into 5 whole square into 10 power minus 4. After doing the whole calculation, you will get the answer as 2.5 and 2 ampere meter. Let's go for our next question. Question number 2 is from your energy chapter. Energy E is equals to h nu, energy of a photon. So A is fourth option. Diffraction and interference, let's keep this aside. Lambda h by p, de Broglie wavelength. You obviously know it's about lambda. So that should be option 1. Compton effect, we know that it's about particle nature, wave nature. Particle nature. So this has to be 2 and the rest is 3. 4, 3, 1, 2. So correct option 1. Let's go for the next question. This is from semiconductors. When you have a diode, you have to know whether the diode is in positive or forward bias or negative, reverse bias. Positive terminal and negative terminals. Positive is connected to positive. Here also positive and negative. So the remaining two are in reverse bias. You have to eliminate those two. So you are left with 4 and 5 and you have total voltage 10. You have to find out the equivalent resistance of 4 and 5. They are connected in parallel. What is the formula of parallel? Resistance product by sum. So product of these two, 20 divided by 9. Then you have to go with V is equals to IR. To find out I, put the V value as 10 and I equivalent here, you can find out the value of current. How much is the value of current? You're getting it as 15 by 2. And the next question is from units and dimensions. In units and dimensions, speed of the light in vacuum is taken as unity, which means C value is 1 meter per second. Light takes a time of 6 minutes 40 seconds. We have to convert into one particular unit now. Which unit do we convert? SI unit. So seconds. 1 minute 60 seconds plus extra 40 seconds. 36 plus 4. 400 seconds is the time. If you know speed and if you know time, how will you find the distance between? Distance formula is S is equal to UT, D is equal to CT. Multiply these two, you will get the answer as 400. The next question is from kinematics, particularly motion in a straight line. So in this, the following plots show the variation of velocity, not speed, with time. The ball is thrown vertically upwards. It goes up and again it comes down. In the first part of the motion, when the particle is projected up, speed or velocity. Speed, magnitude will keep on decreasing. Here the magnitude keeps on increasing. Now, if you focus the acceleration, we take that usually as minus g. So you have to know the slope of the VT curve need to be negative. So slope is negative, it has to be a straight line because acceleration due to gravity is constant. Same the slope of this graph. And then you have the velocity decreasing and then velocity increasing. Here the velocity decreasing becomes zero, and then upward direction we know it's positive, downwards is negative. That's the reason why the acceleration is negative. So later on velocity will become negative. So C is the right answer. So correct option is option 1. Let's go for the next question. Units and measurements, or we can say experimental physics. Vernier calibers. He gave you 20 VSD equals to 16 MSD. Most of the questions were our previous PYQ questions. I think we have seen this in previous PYQ NEET question also multiple times. When 20 VSD is coinciding with 16 MSD, where each MSD is how much? 1 millimeter. You can find out VSD by MSD values. As you know, formula of least count, which he is asking us to find out. Least count formula is 1 main scale division minus 1 vernier scale division. Main scale division minus vernier scale division. I know the units of main scale division, but I don't know about vernier scale. So I will convert VSD in terms of MSD. So it's nothing but 16 by 20 MSD. So if I take common MSD, 1 minus 16 by 20 will give you 4 by 20 main scale divisions, which is 1 by 5. 1 main scale division is how much? 1 millimeter, which he gave you, gives you 0.2 millimeters, or if you are using centimeters, it is nothing but 0.02 centimeters. Correct option 3. Let's go with the next question. Alternating currents in the chapter, it contains a resistance R, capacitance, inductance, everything is connected in series, and the resonance frequency of the circuit. Series LCR circuit question, resonance frequencies XL and XC both has to be same. We know the formula of XL and XC. If you find the frequency, you get the formula of resonance frequency as 1 by 2 pi under root LC. So 1 by 2 pi under root L value is how much? 1 millihenry. So you need to convert that into its SI units, and 0.1 into 10 power minus 6 because he mentioned it as 3 microamperes. So microfarad is 10 power minus 6. If I convert and solve all these terms, 10 power minus 3 minus 6 minus 1, 10 power minus 10, 10 power minus 5, because I have a root. So it's 10 power 5 by 2 pi, and after solving I get an answer close to 0.1591 into 10 power 5. If I take a fraction of that, which is option number 3. Let's move for the next question. This question is from moving charges and magnetic. So here, if I have a wire, for a wire, if the distance is A, radius is given as A, inside, if you have a distance small r or x, let me take it as x. So outside, let's say it is at a distance smaller. What is the formula for magnetic field when it is inside? We used to use Ampere's circuital law, right? Ampere's circuital law, which is integration B dot dl bar is equals to mu naught into I, where dl is 2 pi r or 2 pi x based on the question, and we used to write current of the small circuit of radius x. So we have to write this as total current per unit area into pi x square into b into 2 pi x. After solving, we're going to get an answer for the magnetic field inside as mu naught I by 2 pi a square into x as inside, and outside is mu naught I by, outside it's going to be r, right? So one of the term gets cancelled, 2 pi r, 2 pi r, small r, so pi 2 pi into outside radius is smaller. So if I am going to find a graph or plot a graph for this, First, it is directly proportional to x. Later on, it is inversely proportional to r. So directly proportional has to be a straight line passing through the center, and inversely proportional has to be a rectangular hyperbola. So the correct option has to be 1. Let's move for current electricity question, where he has given a metal wire. Total wire resistance is 4 meters. Now this total wire is made into how many parts? When it's made in the form of a square, each side will be equally 1/4 of it. So each side has to have 1/4 of its resistance, which is 1 ohm. When each side is 1 ohm, and additionally he connected a 2 ohm resistor here, that means 2 volts across A and C. This is exactly and clearly Wheatstone bridge concept. You have a trick for Wheatstone bridge. When you have equal resistances, it can be any other resistor here in between. I'm not going to talk. If all the resistors are R and I have a battery connected in between these two, equivalent resistance is also R. Because current will pass through series, and then parallel. Remember the same concept, we have a similar formula in terms of this in capacitors also, series and parallel. So if your equivalent resistance is R, which is what? Voltage difference V is equal to IR. So I will be and answer is 1. Your next question is about nucleus. So nuclear density is mentioned, mass is also mentioned. What is the mass number? He also gave you R naught 4 pi. He's expecting you to solve using the same concept. Density is equals to mass by volume. Volume of a sphere 4 by 3 pi r cube. But R is equals to R into A power 1 by 3. So if I substitute it here, I'm going to get the answer 4 pi small capital R naught cube into A as my density. So if you want to find out what is A, substitute all the remaining answers. You will get an answer for A close to 12.0. But there is too much calculation involved here, right? I guess you also might have felt the same. So I'm coming here with a small trick or a small formula. If at all you want to find out what is its mass number, you can write the mass of the nuclei and its mass of each nucleus. So, if you have seen the mass here, 19.926 into 10 power minus 27 divided by 1.66 into 10 power minus 27. These terms easily gets cancelled, and approximately if you take this as 20 and this as 1.6, you know that it's going to be 100 multiplied by 1 by 8. We know 1 by 8 is 0.125. Easy fraction. I told you in the last three paper discussion that remember certain fractions 1 by 2, 1 by 3, 1 by 4. So 1 by 8 is such a fraction, 0.125. You multiply 12. Since this number is smaller, it should be less than 0.125. So it has to be 12, which is option number 1. Let's move for the next question. A rectangular wire loop of size 8 centimeters and 3 centimeters with a small cut. Let's first plot this rectangular loop. He has given you a small side 3 centimeters and a large side 8 centimeters, and it's moving in a uniform magnetic field of 0.3 Tesla directed normal to the plane, perpendicular. It can be cross, it can be dot, but it's normal. EMF developed across the cut if the velocity is, he gave you velocity 2 centimeter per second. Focus on the unit, he gave you centimeter per second. Its direction normal to the shorter side. This is the shorter side, perpendicular to it is in this way. You know what? This is an exact exercise question that we have in the NCRT textbook. If I'm not wrong, it is from exercise and question number is 6.4. And most of the paper questions are also the NCRT liners and something so easy to attempt. So now if you have velocity, magnetic field and its length, you can find the EMF formula, which is the EVL sine theta, theta is going to be 90, mutually perpendicular. So simply BVL. B value is 0.3 into 2 into 10 to the power of minus 2 and its length is 3 centimeters. I have to take the smaller one. You're going to get an answer equal to 1.8 into 10 power minus 4. But for example, we have two rods here. Did he give you any other option with 2 into 1.8 into minus 4? No. You just have to come to a conclusion or an assumption that only half part of it is inside the magnetic field and the remaining part which has a cut. Center, there is a small cut and the EMF across these two which we have to find out. That is outside maybe. A galvanometer, current electricity or experimental physics, whichever you are thinking. Resistance of the galvanometer is given. Full scale deflection of the current for a galvanometer. Try to identify what are the variables so that you can find out the formula. Shunt resistance. Such a simple case. We all know that a shunt resistance is connected in terms in parallel with a galvanometer so that I can have a galvanometer which is connected in series with the in parallel with the shunt which will convert into a whole ammeter. Now in this case both are parallel. In parallel is constant voltage. V is equals to IR. Current dividing series, it goes here as Ig, so here I minus Ig. So Ig into G is equals to I minus Ig into shunt resistance. From this you can find out the shunt resistance as Ig into G by I minus Ig, or there is a short trick to this question which is shunt resistance is equals to G by N minus 1 where your N value is I by Ig. But if you still don't want to do that, you can go with procedure. Ig into G is nothing but Ig is small value basically. G is 100. Ig by G is going to give you, which is almost close to I. You can just directly take I value only. I by Ig. So I is how much? 10. Ig 10 power minus 3. I minus Ig is going to give you the same 10. So using this, you can find out what is the shunt. If I solve, I'm going to get 0.01. 1 0 goes here, 10 power minus 2. Let's move to the next question. Young's double slit experiment, so obviously it's from wave optics. If you have wavelength lambda, then its path difference is lambda, same as wavelength, and he gave you its double slit experiment intensity value as k units. As soon as it is from wave optics, we all know the formula of intensity. I is equals to I naught cos square phi by 2. And we also know what is the phase difference and path difference relation. Phi is equal to k delta x, or when phi is equals to 2 pi by lambda into delta x is again lambda. So you are going to get your phase difference as 2 pi in the first case that he has mentioned. So in the first case, I is equals to I naught into cos square. 2 pi by 2 is cos pi, then this is going to be 1. And then you have your intensity which already he gave as k. When you know the value of I naught as k, now it's your turn to find out the answer. What's the second part? Intensity when the path difference is equals to lambda by 3. So intensity is equals to I naught into cos square phi, which is 2 pi by lambda into path difference. So 2 pi by lambda into lambda by 3 whole divided by 2. So all the terms here gets cancelled and you are left with only pi by 3 inside cos. What is pi by 3? 60, which gives you 1 by 2. If you put a square to it, I naught value divided by 4, which I know that as k. So your answer is going to be k by 4. So correct answer is option number 1. Let's move to the next question. The magnitude and direction of the acceleration produced in a body of mass 5 kg and two mutually perpendicular forces are acting on it. Let's say there is a mass of 5 kg. I am exerting a 6 Newton force and an 8 Newton force. We all know a standard trick, a standard triangle, which is 3, 4, 5. When two sides are 3 and 4, oppositely the other side has to be 5. So one is 6 and the other one is 8. So its resultant is going to be 10. You don't have to solve that. Everything you just have to multiply it with 2, or you can use under root f1 square plus f2 square, whichever you feel is convenient. But this is the trick we use to find the answer fast. Now, if this is the total force 10, what is the formula of acceleration? F is equal to ma, so A is equal to F by m. 10 by 5, 2. How many 2 options are there? First 3 options are 2. So, you're going to eliminate this fourth option, and then what is the angle? How do you find the angle? Let's assume this is theta and this is alpha. What is theta? Opposite by adjacent. Tan theta is equals to, opposite is 8. So 4 by 3. Whereas tan alpha is what? Opposite, 3 by 4. Now he said tan inverse 3 by 4 with 6. No, that's not making it with 6. So tan inverse 3 by 4 is making with 8. Right answer is option 2. Last question, which is the 15th question that I am going to discuss for you all. And post this, I'll be handing over to Asit sir. So let's finish this fast. I have all the 4 connected in series, each having a capacitance 10 microfarad, and C5 is again 2.5 microfarad. When each one is 10, what is the formula or a trick for capacitance connected in series? Effective capacitance, if they are all identical, the trick is divided by how many number of capacitors. 4 out 10 divided by 4, your answer is going to be 2.5 microfarad. 2.5 microfarad divided by again. Below this, one more 2.5 microfarad is connected in parallel. And below you have a voltage of 50 volts. If you have your voltage and the equivalent capacitance, when they are connected in parallel, it is just simply added. 2.5 plus 2.5 is giving you 5 microfarad, which he asked here. So 5 microfarad, you have 3 options. He asked you to find the charge on each capacitor. What is the formula of charge? Q is equal to CV. We know, fortunately, above and below both have same capacitance. 2.5, 2.5. Let's find it for C5. Q is equal to CV. 2.5 into, its voltage is 50 volts. So how much is the answer? 125 microcoulombs for all the capacitors. So right answer has to be option 1. So let's hand over this to Asit sir. Please continue with the remaining questions.
Hello everyone. Very good evening to everyone. I hope you all have done very good in exam, right? So let's discuss the remaining problems of the physics. So next 15 question I am going to discuss. Then after me, Jate sir will come and he will discuss the problems, right? Okay. So as we have observed the paper, so there are few questions. We can say in that there are few calculations is needed, right? So that's why we can say in few questions you might be feel that it's a little bit lengthy problem, right? But in general, physics was okay. We can say it's a moderate to difficult, and we are saying it difficult on the basis of that it's a lengthy, right? Otherwise, the questions is not that much tough like the last year NEET exam, right? Okay. So let's go here with the question number first over here, right? Just a moment. Yeah. So in this one, let's move over here. Yeah. So this is the first problem we have, right? So this is from the current electricity. Let's have a look on this. So they are saying in a meter base, in a meter base, this is the given. They are saying there is a cell E and galvanometer Z we have. If we interchange their positions, then what will observe in this, right? So they have just given this observation. So meter base is nothing. You can say it's a simple Wheatstone base, right? So in Wheatstone base, if we are interchanging the position of cell and galvanometer,
there will be no any difference. So we can say we'll still get the both right sided and left sided deflection at the balance point, no deflection, right? So we'll go with the fourth option in this one, right? Now let's move on the next problem. This is the next problem we have from the work for energy. In this one, what they are saying, the power of a crane which lift a mass of 1000 kg to a height of 20 meter in 10 seconds. So we know it's a basic formula. The power is equals to work by time. So we can go with that. This is M, and G and H is given as 20 meter over here, and the time is 10 seconds, right? So they are saying we have to use 9.8, so you can put this as 9.8 over here. So by solving this, you will get the answer as 19.6 kilowatt, right? Now let's move on the next part over here. In this one, this is from the mechanical properties of solids. It's a very easy problem. In this one, you can see they have given the first one, Young modulus. What is Young modulus we have? We know simply that it's F into L by A delta L, right? This is the basic we know. So A is related to second only. There is only one option we have in which A is related to second. So you don't have to check out for the others itself, right? So in this one, we can say the fourth option is correct for us, right? Now let's move on to the next question. So in this question, you can see in a concave lens, ray of light emanating from the object parallel to the principal axis, right? So we have a concave lens over here, and they are saying a ray of light is incident over here, right, parallel to it. So after this, we know it is, we are assuming the para-axial rays over here, right? So in this one, it will appears to just pass from the second focus of this one. This is the focus we have F1, this focus is what, F2. So we can say it should be passes from this second focus, right? But in this one, they have to mention, because in general we are talking about primary focus and secondary focus. So they have to write it properly. It will appears to pass through that focus, right? Okay. So let's move on this one. So we are just giving answer 4 in this one. Now let's move on the next problem. In this one, this is from the rigid body dynamics. What they are saying in this one, we have a ring, and the ring is kept like this. This ring is made up of a wire of length L over here, right? And mass density is M, not mass is M. Mass density is M, right? So in this one, if we are making the ring over here, so length will be 2 pi r, that is equals to L. So radius is equals to L by 2 pi we have, right? Now in this one, what is the moment of inertia of this one? So you can see about this axis we have to find out. So we'll use the parallel axis theorem. What is the moment of inertia about this? This is mr square by 2. It's a diameter over here, right? Let's consider for right now mass is m. So the moment of inertia is equals to mr square by 2 plus m, and this separation is r over here. So we can say this is 3 by 2 mr square. So let's put all the data over here. So this is 3 by 2, m is what? M into L, because m is the linear mass density. Total mass is m into L, right? Into what is R we have, this is, so it's a L square by, it will be 4 pi square, right? So what is the result we have? The result coming out to be 3 by 4 into 2 is 8 pi square, and this is ml cube, right? So which option is correct? We have the first option we have, right? This is the correct we have. Now let's move on the next question over here. So in this one, what they are saying, each side of a metallic cube of this one and measured to be 9 centimeter, keeping the significant figure. So it's a basic question from the error measurement, right? So how we'll find the density? So we all know the density is simply the mass by volume over here, right? What is the volume? Volume will be A cube over here. So volume is what it's given in centimeter. So we can convert it into standard unit, because it's given kg meter cube. So let's see here in this one, we write it, convert it into meter, it will be 0.09 meter cube over here. So it is coming out to be 0.00729 meter cube over here, right? So we can say density is equals to, mass is given as 5.580 by this is 0.00729 over here. So by solving here, you will get as 7.7, right? The value of x will be 7.7. It will comes about 7.654 something. So it's a continuous, so we are, that's why we are taking it as 7.7 over here, right? Now let's move on the next problem over here. In this one, this is from the wave chapter we have, right? So what they are saying, there is a wave equation is given over here. It is traveling along x-axis. What they are saying, where x and y are in centimeter, right? And t is in second. The phase difference between the oscillatory motion of the 2 points separated by 0.5 meter, and they have given xy in centimeter. So you have to convert this one as well in centimeter. So you can write delta phi equals to k into delta x. You can say delta phi equals to k is what? 0.008 into this is 2 pi as well, into this is 0.5 meter. So you can convert it into centimeter. So you will get as delta phi is equals to 0.85 radian we have. So option number 4 we have. This is the correct we have, right? Okay. Now let's move on the next question. So in this one, this is from the geometrical optics, a basic question we have. In this one, what they are saying, a ray of monochromatic light is passing through the equilateral prism, right? This angle is 60 degree over here, as shown in figure. The refracted ray is parallel to its base. When they are saying it's parallel to its base, so simply we'll understand it's a situation of minimum deviation. So in case of minimum deviation, what we are saying? We are saying the delta is equals to I plus E minus A. So it will becomes I and E will be same. So it's a 2I minus A. I is given as 50. So 2 into 50 minus 60. So we can say we have 40 degree over here. So we can say the option 1 is correct for us, right? Now this is the next question we have from the semiconductor. In this one you can see, so in the circuit showing below, the voltage appearing, so they have given this input voltage and they are asking voltage appear across the diode, not the resistor, right? So these things you have to mind in exam. So about this one we have to apply. So initially when this is positive, so it will be in reverse bias, right? It means it will be same potential. But after this, when it will be in forward bias, so all the potential will be across the resistance. About this, the potential becomes zero, right? So according to this, we have only this option. So second option is correct in this, right? Now, let's move on the next one. This is the problem we have. For a simple pendulum having the time period t, the variation of kinetic energy with the time. So in this problem, you can see they haven't said anything how the particle is starting its motion, either how the particle is moving, right? So we can consider that kinetic energy should be positive, right? It should be have positive value and it should be zero when particle is at extreme position. So there is only one option we have. So this option is correct. Otherwise you can go with that the velocity formula k equals to half mv square. V is a omega cos omega t, right? Okay. Now let's move on the next question over here. So in this question, you can see they have given a resistor. Again, it's a question from the current electricity. So in this paper, we are observing there is a more problem from current electricity, right? So you can see in this one, there is a resistor we have. So this is the resistor we have. Let's consider it R. And they are saying we have connected a battery having internal resistance. So this is 2 ohm and they are saying this is 12 volt we have, and current they are saying it's a 0.6 ampere we have. So what is the potential drop over here? We know it's a E minus IR over here. So this is 12 minus 0.6 into 2. So it is coming out to be 10.8 volt we have. So we can say the option 2 is correct for us, right? Now let's move on the next question. In this one, what they are saying, the amount of work done. What is the amount of work done when we are shifting an object, right? They are not saying anything about kinetic energy, so we have to consider it constant. So what is the work done we have? The work done is equals to simply delta U. So they are given the height equals to the radius. So you cannot go with that mgh, right? So what you have to do? You have to apply the concept of that mgh by 1 plus h by re over here, right? So now in this one, this is mg, h is what? R, and this is 1 plus r by r. So result is coming out to be mg r by 2. So which option is correct? We have the fourth one is correct, right? Now let's move on the next one. So in this problem, what they are saying, they have given an electric heater supplies heat to a heat means it's supplying energy to a system at the rate of this 100 joule per second. If the system performs work at the rate of this one, then they are asking what is the rate of change in internal energy, how it is changing. So simply you can just say it is providing heat over here at dq is equals to dw plus du we have, right? So they have given power, so we can write it dq by dt is equals to dw by dt plus this is du by dt we have. So they are asking about this one. This is they have given 100, this is given 75, plus this is du by dt. So you can see from here this is coming out to be 25. So third option is correct for us, right? Now let's move on the next part over here. In this problem, what they are saying, a room heater is rated. These are the rated value we have, and on this we are just using it, right? So what they are asking, they are asking what will be its power consumption, either consumed power at that voltage. So we know P is proportional to V square. So P rated by P consumption is equals to V rated square by V applied square over here. So what is the P consumption over here? So V applied by V rated square into this is P rated over here. So what is the P consumption we have? It is coming out to be 200 by this is 220 square into this is 400 over here, right? So after solving this, you will get the answer 2 we have, right? Now you can see this one. This is more than 1, this is less than 1 over here, and this is 400, right? So it is coming out to be less than 400 and it will be close to that. So only this option we can take, all are very far away from that one, right? This 400 cannot be there, right? Okay. Now let's move on the next question over here. Let's see here. In this problem, you can see what they are saying. When a ruler falls vertically, 5 different person catches it with different reaction time. What is reaction time? Reaction time is basically, it's a time interval between observation and reaction, right? So let's say a ball is coming down over here, so they are observing. So in their reaction time, ball will fall under the gravity itself, right? And they are saying it's fall vertically. So let's consider anything. For that time over here, let's consider it free fall. So you can go with half zt square as well. So if reaction time will be more, that will travel more, right? So for which reaction time is more? For B reaction time is more. So there is two options with that. So we have to check out for others one. For next one, I think after B there is a E. So E have more time. So fourth option will be correct for this, right? Okay. So we have already covered this 15 question. Now the next 15 question will covered by the jacket, right? Okay. Thank you.
Okay, thank you, Ajit sir, for the opportunity. So let's get started for the last 15 questions in the NEET. And what we have seen in the NEET, as the previous sir have mentioned, that these questions were simple. There were a bit of a calculation somewhere, but overall it's kind of like an easy thing. So let's begin with the question number 30. 31. So listen carefully. Here, the question is mentioning about the capacitance, and we got here capacitance is equals to C, that is 200 into 10 to the power minus 12, that's a picofarad, and then we got here voltage, which is going to be 100 volt. Now they're asking if it is connected with an uncharged capacitor and then they are sharing the charge, how much amount of electrical energy will be lost into the process. So answer is going to be much easier. The delta energy which is going to be lost is half of the initial energy we got, which is half CV square. This you learn because when you have two capacitance same and you're basically connecting them, what happen is these two are going to get total voltage as V by 2, and then when you do the calculation, this is going to be concluded. So here we are going to get delta E is equals to half into another half into C value, which is going to be 200 into 10 to the power minus 12, and then V square, which is going to be here right there, 100 volt. So 100 volt whole square. Now if you simplify this thing, it is going to be come out to be 0.5 into 10 to the power minus 6, which is going to be second option. Let's move on to the next question. The next question is, it's very simple. Let me tell you, it's very simple. You don't even have to do the calculation. Just read this. The time taken to complete the 30 oscillation in 60 seconds, meaning time period is 60 divided by 30. And the moment you get this thing is 2 seconds, you should realize it's a second pendulum, and second pendulum means its time period is going to be, well, 2 seconds, which means length automatically is going to be approximately 1 meter right away. Moving on to the next question. Next question is from alternating current. It says that the alternating current 5 ampere, frequency 60 hertz, how long will the current starting from 0 to take to reach the peak value? First thing first, the graph of the current will look like this. If this thing is T, that thing is T by 2, then the peak voltage is going to be at T by 4, from 0 to T by 4. So time here is gonna be T by 4. Now what is T value? T is going to be 1 upon frequency, which is 1 upon 60. So this is going to be 1 upon 4 multiplied with 1 upon 60, which is going to be 1 upon 240. Right this way. That's the first option. Now moving on to the next question. Now, next question is not calculative, it's basically the assertion reason. It says interference and diffraction of the light is redistributed. Okay. Now, here they said As there no gain in loss of the energy, these phenomena are consistent with the principle of conservation of energy. Definitely. There is no loss of energy happening whenever we do any one of these things, whether it is young double slit experiment or single slit experiment. Next thing is the diffraction and interference characters exhibit only by light wave. That is highly wrong, because it's not just electromagnetic wave who gives that thing, it also the sound wave, it is also the other wave as well. So basically it is applicable for all waves, which means the B is going to be false, A is going to be true. First option is correct. Moving on to the next question. The next question says there is a box of 15 kg which is having a coefficient of friction with the platform like this. This is 15 kg and it has mu value right here equals to 0.12. They are saying that the maximum acceleration for the trolley, for which, meaning basically the platform, such that it will not slip. So here we want to apply something like a force which will give the entire thing acceleration such that it will not slip. Now, not to slip here means the force must be balanced. The MA part must be balanced, or should I write it this way? The friction force on the block is going to be forward, which is going to give the acceleration, or should you say the pseudo force Ma will be balanced by mu mg. So just do that, and just like that you would be getting acceleration is equals to mu g, which is simply means multiply these two data, and that would be option 4. Now let's move on to the next question. Next question is sum of the kinetic energy and potential energy of the simple pendulum is 0.2, which means half k a square, or should I say half m omega square a square. Basically both are same. The speed of the pendulum bob at the equilibrium position approximately. Now at equilibrium position the speed is maximum, which is a omega. That's a max speed. Okay, so we get here half m v square. Half m here is 20 gram, which is 20 into 10 power minus 3, and v square is going to be, we need to find it, must be equals to 2 into 10 to the power minus 2. Now if you simplify this thing, you'll get v square is equals to 2, which means root 2. That means we're looking for 1.41. Moving on to the next question. The next question is not calculative. Again, we got sort of like a statement checking question here. So let's see. It says the volume of the nuclei is proportional to a to the power minus a to the power 1 by 3. So wrong. It was supposed to be radius. So A is wrong. Next is B is volume nuclear proportional to A. That's correct. Difference between the mass and the atom and its nuclei. No, 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 not the nuclei. It is supposed to be the constituents, which means C is wrong and automatically D is correct. That means A and C are going to be wrong, and B and D are correct. First option. Moving on to the next, the angular speed. First of all, this question from the circular motion, it's actually the kinematics part. The only thing about this question is this, it's tough. I mean, in the terms of calculation, it's a bit bigger. So let's see here. It says we got omega. So let's call it omega final as 12 rpm, which is gonna be multiplied with 2 pi by 60 in order to get this thing in terms of radian per second. That came out to be 40 pi, and then omega initial is going to be 600 into 2 pi divided by 60, which is going to be 20 pi. Now they have given the time, which is going to be 10 seconds. First we need to calculate is alpha, which is going to be final minus initial divided by time t. So omega final is going to be 40 pi whole square minus 20 pi whole square divided by time, which is 10, which is going to be, I mean, you can solve it here. Oh, sorry, my mistake. It is not supposed to have any whole square right here. So if you solve it, which is going to be 20 pi divided by 10, 2 pi, and then you are going to calculate the theta, which is going to be omega f square minus omega initial square divided by 2 alpha. You will get this thing as 300 pi, which is 150 revolution. Now, moving on to the next question is submarine is designed to withstand an absolute pressure of 100 atm. Okay, so first thing first, at surface there is already a P naught which is 1 atm. Now you are going to get up to a depth H which will give the pressure P naught plus OGH, and that's supposed to be 100 atm. Now this thing is already 1 atm. That means rho GH is going to be 99 atm. That means 10 to the power 3 into 10 into h, which is going to be 9, 9 into 10 to the power 5. Simplify that, that is going to be 990 meter right away. Moving on to the next. Next is, well, statement type question. So listen carefully. It says like bunch of electromagnetic waves is given and you need to figure out the things. First thing first, very easy one. Gamma rays radioactive, like it's literally the name here. Then visible light is related to the electrons because it does have something called Balmer series. Next, microwave is related to these two walls. And infrared, well, if you did the chemistry well, you can right away tell it is going to be vibration of the atoms and molecules. So by looking at this thing, we are looking at A4, B first, and C second, and D third. Do we have that? Yes. A fourth, B first, and C second, and D third. Now, moving on to the next question, it says which of the following is correct. Now, you know what? Let's do something reverse. How about we find the incorrect version here? So, incorrect version is going to be inside the conductor, the electrostatic field is zero. That is correct. The electrical field at the surface of the charge conductor does not depend in its charge density. That is highly wrong because we know electrical field is equals to something like a sigma divided by E. Sigma divided by epsilon naught. Now moving on to the next, the interior of the charge conductor having excess charge, that is wrong. And at the surface, the charge conducting electric field must be normal to the surface at every point. That is correct. And the electrostatic potential is zero everywhere inside the charged conductor. Look, that is wrong because we know the potential is going to be constant. The electrical field is going to be zero. So that is wrong. So at the very least, I'm confirm about the B and E part. Let's read that C part once again. The interior of the charged conductor can have no access. Oh, sorry about that. No access. Then in that case, it's a correct one. No access. Now, after that, checking out, let's see which option is going to be correct here. So we got A, that is correct, C, that is correct, and D, A, C, D only. Moving on to the next. For a metal work function 6.6, which of the following wavelength incident radiation does not give rise to this thing? Look, simple. Hc by lambda, energy. 12400 by lambda is gonna be 6.6 electron volt. Simplify, it will come out around something like a 180 something because we are not doing the entire calculation and you don't really have to do it, believe me. If you do this, you will realize anything bigger than that gets less energy, 200 right away. Moving on to the first excited state of the hydrogen, the energy of its electron is given. Look, write this, gives the hint it's n equals to 2. Then the radial distance will be how much? Easy peasy question. Radius is equals to 0.5 angstrom into n square by z. Z is 1 because hydrogen. N is going to be 2. By the way, it's a 5.3 approximately. If you solve it, you are going to get the answer either way. Look, you don't really need to solve it, really, believe me, because this right here, this tells the entire thing. You're going to get fourth option correct. Moving on, to again the statement type question we got, and let me tell you something, even if you don't know the concept, it's so damn easy. It says forward bias and next it says it's a reverse saturation current. That's right, there is wrong. That's it, you don't even have to read the statement. But if you do, the forward bias voltage PN junction diode increases above the certain threshold voltage. Basically, they are talking about the knee cap, the knee voltage there. and then it is going to be obviously forward bias, not a reverse one. So B is false, A is correct. So we are gonna be having second option as correct here. Moving on to the next one and the last question. Now, last question came from the kinetic theory of gases. So, what we have this question: the flask contain an argon and chlorine of this with mass, but let me tell you, it's useless because they're asking VRMS. VRMS formula is square root of 3 kT by m. Look, I prefer K, which is Boltzmann constant. You can write R here. Just be careful with the molecular mass, molar mass. Now here for argon, Ar, and then we got chlorine. So basically, look, 3 kT is gonna be same for both of them. Both are 27 degrees Celsius. Cancel out. So mass is gonna be important here. 70 goes here for chlorine, and then 40 goes here, which is gonna be square root of 7 by 4, which is gonna be root 7 by 2. And with that, our question has like all of them completed. So at the end, this is from a message from myself, Zakir Hussain, physics faculty at Resonance, that the NEET paper, especially the physics portion, came to the same thing as the level it was supposed to be. So it's gonna be simple. Just read the formula, apply the formula, skip the question which are too calculated, and boom, that's all you need to do. And thank you for listening. Let's meet in the next video.